Dear Mr. Dorin Grecean, Prime Minister of the Republic of Moldova, dear Professor Dorin Grecean, Director of the Technical University of Moldova, His Excellency Mr. Yukiro Yamada, Ambassador of Japan to the Republic of Moldova, dear Professor Randy Sherman, University of California, Berkeley, Nobel Prize laureate, dear Professor Nicolae Palikarakis, International Federation for Medical and Biological Engineering, dear Professor Emil Japan, Rector of the State University of Medicine and Pharmacy, Nicolae Testemitanu, dear Professor Dr. Shonta, co-chairman of the International Conference on Nanotechnologies and Biomedical Engineering. Dear attendees, ladies and gentlemen, good morning. It's a nice day in Moldova, 20th of September, good weather, and on behalf of the International Advisory Committee, on behalf of the program and organization committees, I would like to welcome you to the 6th edition of the International Conference on Nanotechnologies and Biomedical Engineering. So we started to organize this series of conferences in 2011, 12 years ago. With the time, we gained experience, and more people, more and more well-known researchers, professors from many countries of the world started to attend this conference. And I would like to say that indeed it became a very strong international meeting where researchers from many countries meet, discuss, exchange opinions, start to prepare and to promote joint projects. And Moldova became stronger also through the organization of this conference. Of course, thank you very much for your acceptance to attend the conference in spite of the fact that the coronavirus is still in the air and there is a war in our region, in the neighboring country. In spite of all these barriers, you are here. And this reflects your high interest to the scientific research in general and interest in your desire to meet our researchers and to discuss joint results and to start new joint projects. Ladies and gentlemen, it's my pleasure to give the floor to Mr. Dorin Grecean, Prime Minister of the Republic of Moldova, who will come with a welcome message. Excellency, you have the floor.
Thank you very much. Look, I'm extremely honored to be among the best intellectuals, not only from the Republic of Moldova, but from elsewhere on this planet. I'm only very happy to welcome our scientists, but also the scientists from abroad. And I know that Moldova can contribute, and now with globalization, it's even more important to have this collaboration. So I'm only very happy to see this conference having taken place every year in Chisinau. I'm also very glad that we are aligned with the future. The way I see the future, which is becoming extreme actually, is that we have to advance in 4 fields. The first is the material science. And here we are looking into more and more resilient, flexible, and other types of materials that allow us to prosper. The other thing in the material science is how we can build things and materials not only on our planet but elsewhere as well. And here we have progress. The second field is the life sciences. And here the people want to live longer, want to live healthier, and the science has an answer to that. The third field is the computer science. And here people want to have the replication of our brains, how we think, and this is our brains is very efficient, and correspondingly we are looking into how to replicate this. Machine learning is machine learning, artificial intelligence is still something to be achieved. And the fourth field is the human talent. The human talent at its best use and best development. I'm very happy to see that the conference addresses directly the first two fields and contributes with the other two fields indirectly. I wish you success. I saw the program. It's very intensive. I wish you all the success, and I'm sure that at a certain point in time, some industrial or human life applications will come out of the work of today's conference. Thank you very much.
Thank you. Ladies and gentlemen, it's my pleasure to give the floor to Professor Vera Bostan, Director of the Technical University of Moldova. Please.
Distinguished participants, ladies and gentlemen, a very good morning to you and welcome to Chisinau. And we are very honored to be co-organizers of this important conference, a conference that already is marked by high achievements, bringing together scientists not only from Moldova but throughout the world. I'm very also honored to have here with us a remarkable scientist, Professor Andy Sheckman from University of California at Berkeley, whose roots are from Bessarabia. I'm very happy to have old friends again, despite all the problems we have in the regions, who came to Chisinau. I would like to thank the organizing committee and the scientific committee for their dedication and work and competence in setting up this forum, and also to thank in a special way Professor Victor Shontea, who is one of the founder fathers of this conference and whose efforts actually brought us here in Chisinau today. Dear colleagues, I'm sure that this conference will contribute to the development of the research not only in Moldova but throughout the world. And this conference is a special one because, as Mr. Prime Minister mentioned, here we have two important fields which today represent one of the driving motors of change. Those fields are nanotechnologies and biomedical engineering. And in the same manner as semiconductor technology reshaped the previous 70, 80 years, today I'm very confident that nanotechnologies, material science, and biomedical engineering, biomedicine in general, will reshape the years to come. I wish you success in the works of this conference, and I'm hoping that this conference will contribute to the development of science in the Republic of Moldova, and not only will enhance our cooperation and will lead to new projects. I wish you good luck, and thank you very much for your attendance of this important forum.
Thank you. Ladies and gentlemen, I would like to give the floor to His Excellency, Mr. Nicolae Yamada, Ambassador of Japan to the Republic of Moldova. We have many guests, many professors from Japan. It's a tradition already that we have excellent teams attendees from Japan. So, please, you have the floor.
Good morning, everybody. Thank you, dear Prime Minister. Thank you, dear President of the Academy of Sciences. Thank you, and good morning. And well, my name is Yoichiro Yamada. I'm ambassador of Japan to the Republic of Moldova. I came here about 9 months ago and trying to understand what this country's strengths are and how, as ambassador, I can be a bridge between the Republic of Moldova and Japan. And then one of the things that I found was that the Moldova is very strong in nanotechnology. I was really surprised and impressed when I paid a visit to the Technical University of Moldova, and then it was shown around to be acquainted with the achievements that university has made. And I'm really honored to note that there are distinguished professors from Japan. And then this is certainly an area most advanced in making humanity closer to a greater, higher levels of prosperity. Talking about nano, there was a nano satellite launched into orbit, which was a small satellite manufactured by the Technical University of Moldova last year. And then the launch pad was a Japanese spacecraft. So here was another example where nano brought Moldova and Japan together. But well, science is an area where international cooperation, not just bilateral cooperation, but really multilateral international cooperation makes sense and has made sense for all these years. And it's really exciting to see so many distinguished researchers, professors from all over the world to be present here, and I'm very honored to be invited to this podium today. Thank you.
Thank you, Your Excellency. Ladies and gentlemen, I'm honored to give the floor to Professor Eddie Sheldman from the University of California, Berkeley, Nobel Prize laureate in medicine, a good friend of the Republic of Moldova, honorary member of the Academy of Sciences of Moldova, and Dr. Honoris Causa of the State University of Medicine and Pharmacy, Testemitanu. Professor Sheldman is in the Republic of Moldova starting from Sunday evening, so already has some impressions, and actually, as far as I know, positive impressions, and I am happy to give him the floor.
Thank you, Professor Chisinau. Ladies and gentlemen, I'm delighted to be here. This is now my third day visiting Moldova, though the trip has been long planned. Professor Chisinau first approached me several years ago after he learned that in the biography that I posted online, I said that my grandparents came from Bessarabia. And so naturally there was an affinity, and I'm delighted that he reached out to me. My family back in the States is quite excited about learning about this trip, and I'll be the first person to return to this area in nearly 100 years, because my grandparents emigrated in 1927 from a then village of Lipkan, which I will return to see tomorrow, so I'm very excited about that. I've had wonderful opportunities in the first 2 days to visit the Technical University and at the Medical University, and to visit with faculty and share my thoughts. I'm impressed, just in the brief time that I've been here, at the urgent desire to advance as a scientific society emerging from years of control by the Soviet Union and still the shadow of Russia now being put aside. Moldova has a tremendous opportunity to exploit the natural talent of its young people, but it must invest even more in science so that the young people who are trained in universities here have an opportunity and are encouraged to remain to develop not only the intellectual basis of this country, but also the industrial base. I'm eager to help. We visited with a parliamentarian yesterday, and I see a path forward, and I'm eager to share my own research with you this morning. So I'm delighted to be here and look forward to meeting more of you.
Thank you. Ladies and gentlemen, it's my pleasure to give the floor to Professor Nicolae of the International Federation for Medical and Biological Engineering. Professor Palikarakis is also a friend of the Republic of Moldova. Please. Doctor honoris causa of the Technical University. Please, you have the floor.
Thank you very much. It is my honor to be in this Congress again. I think that it is in the majority of the organization of this conference I have been present, and this is due to the fact that 15 years ago I met Victor Sontea, and through two big projects of Tempus in biomedical engineering education, we created very strong links. But it's also an honor because it's the first time actually that I'm in a conference that the Prime Minister of the country is present, and I'm so glad and grateful that you recognize the importance of biomedical engineering by your presence here. So today and this time I'm going to address this Congress on behalf of Ratko Majarevic, who is the President of IFMBE, and I have to give you some information about the International Federation of Medical and Biological Engineering. Okay, we have the, okay. So IFMBE, who is the International Federation of Medical and Biological Engineering, has been created more or less 60 years ago with main mission to encourage, support, represent and unify biomedical engineers all over the world in order to support better health applications for the benefit of patients. IFMBE is the largest biomedical engineering organization in the world with many national and international partners. It has been created in 1959 in Paris and has been firstly registered in Paris in 1959, I think. Today we have a very complicated, I would say, but very large organization with its administrative council that Ratko Majarevic is the president right now. There are ordinary members of this council, there are regional groups, representatives in the council participate as well, and also there are 3 divisions of IFMBE representing very critical sub-specialities of biomedical engineering: clinical engineering, health technology, assessment, and digital health, very latest. I had the honor and the chance to be chairman of Clinical Engineering Division and the Health Technology Assessment Division, and there are also some supporting bodies, committees, etc., that support the general function of IFMBE. Here you see a map on the different regions and countries that are represented in IFMBE. IFMBE is part of the IUPESP together with the International Organization of Medical Physics, and through IUPESP it is very well connected to United Nations Organization, to International Science Committee, and also to standardization bodies. As concluded, in order not to be so long, since the start in 60 years, 65 almost years ago, has grown very significantly, and today it represents the most large organization in biomedical engineering. Unifies biomedical engineering community, and as IFMBE, we appreciate the effort by all our members to participate and support these actions. So it is essential that all the societies are supporting the role of and the tasks of IFMBE, and we are very glad and grateful to Professor Sontea and the Moldovan Society of Biomedical Engineering for the support that they give. So thank you very much again. It is my pleasure to be here and address the way that this conference will conclude.
Yeah, thank you. Thank you so much. So I would like to give the floor to Professor Nicolae, Rector of the State University of Medicine and Pharmacy.
Good morning, everybody, dear Mr. Prime Minister, dear guests and colleagues. Biotehnologiile, tehnologiile medicale, ingineria, astăzi este o necesitate. Noi acest lucru l-am analizat și sunt foarte mândru și bucuros că participăm ca coparticipanți la această expoziție. Astăzi, medicina nu poate fi fără tehnologii, fără tehnologii noi. De aceea, eforturile comune și împreună cu domnul rector Bostan avem planuri pentru a dezvolta aceste programe noi, a introduce în sistemul educațional pentru a implementa în sistemul de sănătate în Republica Moldova. Vă mulțumesc toți pentru participare și sunt mândru că avem un număr foarte mare de lucrări la acest eveniment. Mulțumesc!
Professor Nicolae told about the importance of nanotechnology, biomedical engineering, biomedicine in general, and of course he is very happy that attendees from many countries are in the conference hall, and wishes the best to all attendees. So, ladies and gentlemen, I would like to give the floor to Professor Victor Șonte, co-chairman of the International Conference on Nanotechnology and Biomedical Engineering. Professor succint.
Dear colleague, thank you for participation à notre conférence. Am spun câteva cifre. La conferință au fost depuse 145 de lucrări din 33 de țări.
140 papers submitted from 33 countries.
Deci, astăzi noi avem aici mai multe țări au sosit, încă astăzi mai sosesc, dar la conferință ne așteptăm în zilele acestea grele, când suntem să mai viziteze Moldova, ne așteptăm undeva la 85-90 de persoane din toate țările lumii.
So we have between 85 and 90 attendees from many countries of the world. So some experts are still on their flights and they are waiting for them.
Eu vreau să mulțumesc Comitetului de Organizare și în primul rând domnul Sergiu Răilean. Nu e aici.
Ok, altă dată.
Deci, și aș vrea să spun că noi lucrăm mai departe fix după programul conferinței. Noi nu avem nicio abatere, deci încercăm să lucrăm după programul conferinței.
Thank you. So we will continue our work according to the program. No changes in the program. That's very important. Do not leave the conference space because we will work according to the program that was elaborated.
Și știind că mulți din dumneavoastră o să viziteze Cricova.
Many of you will visit Cricova.
Deci asta este valoarea națională a țării noastre.
Yeah, so Nicolae is a well-known place in the Republic of Moldova as well in the world.
Eu vă previn că acolo sunt 14 grade Celsius.
14 degrees on the ground in Nicolae.
Și să vă îmbrăcați mai bine.
So you should have the cloak with you.
Și deci vă doresc succes și vreau să prezint că noi asta este un forum științific, dar fiecare forum științific se evaluează și se acreditează. Și astăzi noi îl avem pe președintele Comisiei de Acreditare și Cercetare din Republica Moldova, un participant regular la conferințele noastre, domnul Andrei Chiciuc. Care îl salutăm.
Professor Andrei Chiciuc, evaluate our presentations. Thank you so much. So, ladies and gentlemen, we will pass now to the realization of the program, and I would like to ask Professor Palikarakis and Professor Rainer Adelu to take the floor and to start the first plenary session, and I hope that the presentations are there. Thank you, Excellency, for your presence here and for your welcome messages. And now let Professors Palikarakis and Adelu come here. Thank you so much.
It's my honor to co-chair this session with Professor Rainer Adelu. And I have the pleasure to introduce you the first speaker. This is Randy Sigman from the Department of Biology and Cell Biology from the University of California, Berkeley, US. Sigman, the floor is yours.
Thank you. Well, good morning again, and I'm delighted to be here. The title of the talk that I gave for the program is rather more general, but I thought since this is a specialty symposium that I would not just talk about old things, but rather about new things. And so I want to share with you a subject that I think will be of interest to people who study nanomaterials and may be interested in biotechnology. And that is a process that all cells use to export not just protein molecules from the cells, such as hormones, but rather all cells also manufacture little particles, vesicles that have a bilayer membrane surrounding them. In fact, in our bodies, there are billions of these little vesicles called extracellular vesicles that float around and which have captured in their production molecules, both protein and RNA molecules, that were present in the cells of origin. In fact, it's known that tumors produce extracellular vesicles more abundantly than normal cells, and so there is a great promise in using, in liquid biopsies, for instance, in small blood plasma samples, it may be possible to detect tumor antigens on extracellular vesicles that are produced even at the earliest stages of cancer before metastasis. And we know that if such detection can be done sensitively at the earliest stages, then chemical or immunological or surgical intervention is much more successful. So the hope is that these little markers, these little vesicles, can be used as a source of biomarkers to evaluate stage 1 cancers, and I serve on in an advisory capacity for a biotech company in the US that has devised a very sensitive way of detecting 3 different tumor antigens that are present on individual particles in plasma, blood plasma samples taken from very early stage ovarian cancer. So if we can detect that in stage 1 ovarian cancer, the prospects of more effective treatment are really quite dramatic. So, but I'm going to tell you about the basic work in my lab to explore how these vesicles are produced and for what purpose. Are they really produced to serve as a means of communication between cells, or are they produced for other reasons? So let's dig into what we've been looking at. So first, a summary of what is known about the production of two populations of extracellular vesicles that are made by human cells. All of our experiments are done with cells grown in tissue culture. And for the purpose of this discussion, we've used two different cell lines. One is a non-transformed human embryonic kidney cell line that is used by a number of laboratories around the world, but it's non-cancerous. And another is a breast cancer cell line that is quite robust and which produces these extracellular vesicles in abundance. So we know from studies in many laboratories, including my own, that there are two populations of such vesicles. There are vesicles, can this be seen? I can see it on my finger, but I can't see it on the screen. Can you see this red dot? Well, no, that's not what I want. Okay, well, it may prompt other people too. So if you focus on the cell on the left, there are small vesicles that pinch from the cell perimeter. These are called microvesicles or shedded vesicles. And when they pinch from the cell surface, they capture molecules from the cytoplasm that become enclosed within membranes. This is, by the way that envelope viruses like HIV are produced. They pinch and they capture their nucleic acid core and then are shed into the medium. Another method that produces these vesicles is shown just to the right. It's a process that relies on a pathway within the cell called endocytosis, where membranes within the cell accumulate vesicles in their interior to create a structure that I've abbreviated MVB for multivesicular bodies. These MVBs deliver their content to the lysosome, where molecules are degraded, but they also can deliver their content by membrane fusion to the cell surface, where vesicles are expelled, and these vesicles are specialized vesicles. They're called exosomes. So you may have heard that term, exosomes. That is one of two populations of such vesicles. Now here's a cartoon that illustrates the composition of exosomes. They are simply small bilayer enclosed particles or vesicles. They have membrane proteins in them. I'll talk about one particular one that spans the membrane 4 times. They have constituents in their soluble interior, including small RNA molecules. And they have phospholipids in the bilayer. And the membrane of the vesicle is in part a reflection of the cell from which this vesicle was produced. And so they carry antigens that come from the cells of origin. Thus, those antigens have the possibility of being detected in blood samples. Now, several years ago, a graduate student in my lab, Rima Tomosh-Diaz, devised a way of separating these two populations of vesicles on a buoyant density gradient to separate membranes according to their composition. Membranes that are rich in lipids tend to have a lower buoyant density than membranes that are rich in proteins. And two populations emerge on a linear buoyant density gradient, as you'll see in this slide. So this is an immunoblot of different antigens from these populations. And if you look at the lanes on the left, you'll see representative blots of proteins that are enriched in the low buoyant density population, which we know to arise by pinching from the cell surface. And you'll see on the right a higher buoyant density population of vesicles that are enriched in a protein called CD63. It's an antigen, a polypeptide that crosses the membrane 4 times. So it's an integral membrane protein. Now we know in other experiments that I won't have time to tell you that these two populations of vesicles that can be separated on this gradient have different small RNA molecules. And we found and studied in some detail, but I don't have time to tell you today, that the vesicles that are at the higher density region carry very specifically certain small RNA molecules called microRNAs. And so the surprise is that these vesicles can very selectively capture those small RNAs from the cytoplasm of the donor cell. We're studying how that happens. There are specific RNA binding proteins that are required for that. And we're wondering what purpose that serves. Why would cells go to such trouble to enrich particular microRNAs? We estimate in some cases by as much as 1,000-fold. So that's a subject for another day for discussion. Now, what we wanted to do, and what the subject of my comments over the next 30 minutes, what we really wanted to do was to address in the next experiments whether these vesicles that are being produced so prolifically by all the cells in our body, whether they are means of communication. Because the possibility existed, indeed many people believe, that these vesicles capture certain molecules from a donor cell and then deliver them into the cytoplasm of the target cell. For instance, there's a prominent view that tumors, primary tumors, produce these vesicles, and the vesicles float around in our body and then are taken up by certain other tissues to create what is called a pre-metastatic niche. That would be a site into which cells that come from the primary tumor may migrate to establish a metastasis. So if that's true, and there's some evidence to suggest that, if that's true, that's another way of attacking cancer. If you could block the production of those vesicles so that they couldn't move to another site of the body, you might prevent metastasis. And that becomes a much easier means of curing the cancer in the first place. So there are many really interesting prospects that arise from the study of these vesicles. Well, we were, I have to say, rather skeptical of the evidence that these vesicles can actually communicate effectively. And so a Chinese postdoc in my lab by the name of Songyan Zhang decided to devise a test, a very specific test, that would allow us to measure quantitatively the efficiency with which molecules that are in exosomes can be delivered to the cytoplasm and possibly to the nucleus of a target cell. So here's the strategy that he used. We relied on a protein that I mentioned, the CD63. We know this protein is highly enriched in the membrane of the intracellular organelle called the endosome. And we know it is also very highly enriched in the membrane of the exosome. I showed you the data for that in the previous slide. So we thought, would it be possible to use the presence of this protein to capture the enzyme called Cas9? So you probably all heard about CRISPR-Cas9 and the use of these molecules in genome editing. Cas9 is a DNA endonuclease, and when Cas9 is bound to a particular small RNA molecule, that RNA will convey to the nuclease a very specific target in the genome that would allow the nuclease to make a cut, and then by a process of error-prone repair, you can change the gene into which the cut has been made. So we devised a technique which is outlined here, whereby the Cas9 protein synthesized in the cytoplasm of the donor cell is attached to another protein by way of gene fusion, another protein that's called strep-avidin binding protein. And on the other side, we fused to the CD63 molecule a different protein called strep-avidin. Now, strep-avidin happens to be a biotin binding protein, and it will also, by chance, interact with strep-avidin binding protein. The two proteins will touch and bind to each other in a non-covalent manner. They can be broken in that interaction by simply the addition of exogenous biotin. But in a normal cell, the two will tether to one another, and if the cells also express the small RNA that can be used to guide Cas9 to a target, that RNA will be carried along, interact with strep-avidin CD63, and eventually captured into a vesicle that buds into the interior of the endosome, such that the MVB that I described a couple of slides ago will have a pool of Cas9 and RNA molecules enriched in the interior of this vesicle. Now, when the MVB delivers its content by membrane fusion to the cell surface, the cell will expel lots of exosomes that are now enriched in Cas9 and a guide RNA inside the vesicle. Now, the purpose of this was to produce vesicles that would then potentially be delivered to a target cell where the nuclease could attack a gene in the target cell, and that would be a measure of the release of that enzyme from the interior of the vesicle. And you'll see that that reaction, the release of content of the vesicle, requires that the membrane in the target cell fuses by a membrane fusion process with the vesicle. That will become important in the succeeding slides. So here's just a little evidence that what we claimed we could do, we actually could do. The cells, in this case human embryonic kidney cells, were grown in culture. The supernatant fraction, which had all of these exosomes, was harvested. The vesicles were centrifuged and then applied to a density gradient to allow vesicles that are buoyant to float, to separate them from other particulate matter. The enriched vesicles, which you see in the cartoon by a dark band in this tube, were then removed from the tube. And we evaluated their content by using antibodies in the form of a Western or immunoblot. The first two lanes in this slide on the panel show preparations of vesicles that were collected from cells where Cas9 was not bound to CD63. It was just freely expressed in the cell. Whereas the third lane shows a sample where the Cas9 was tethered by virtue of this streptavidin binding protein streptavidin interaction. And you could see a remarkable difference in the ability to detect by an epitope tag the presence of Cas9 in the vesicle of the third preparation, which was not represented in the vesicles that didn't have Cas9 tethered to CD63. So there was a tremendous enrichment of Cas9 by this trick. Now, of course, at the same time, we could evaluate by PCR the presence of the guide RNA that was expressed in the donor cell. And you can see that in the first two samples where Cas9 was not tethered to CD63, very little RNA was captured by those cells into exosomes, whereas in the sample where Cas9 was tethered to CD63, we found a 15-fold enrichment of the RNA that was bound to Cas9 and delivered into these vesicles. So that worked. Now, of course, that's only half of what we needed to have this assay. The other half was to design an acceptor cell that would take up these vesicles and possibly deliver Cas9 into the nucleus that would be detected by the activity of the Cas9 endonuclease on a gene that we could measure, where we could measure the effects. So here's the technique that Song Yan devised. I won't go into it in detail, but this work has been published if you're interested. What we did was we created in the acceptor cell, in this case of the breast cancer cell line, we created a cassette of genes that had a gene for a very potent enzyme, N-luciferase, an enzyme that can be detected photometrically, because when the presence of ATP in a substrate, that enzyme generates photons, so it's very sensitive and very active enzyme. But the enzyme, the gene that was in this cassette, was crippled by the presence of a stop codon that would prevent the protein, the gene, from being translated into a protein. And it included a single nucleotide deletion mutation that would shift the reading frame of the gene so that it simply couldn't be expressed. Indeed, if you look at this crippled copy under the right circumstances, if you see in the plot on the right, in the first lane, there's no enzyme activity detected. On the other hand, if you introduce the Cas9 and guide RNA into this cell, there is an endonuclease activation by cleavage of the crippled copy, and in the attempt to repair the cleavage by Cas9, the cell uses an error-prone repair process that will overcome the presence of the stop codon and the single nucleotide deletion and restored reading of the gene to make N-luciferase. And you can see on the right that when you express both Cas9 and a guide RNA in this tumor cell line that has the crippled copy of luciferase, you can see a 100-fold increase in the expression of luciferase activity. So the details of this are not important for this discussion, but it's just a very sensitive, accurate reflection of the introduction of Cas9 likely into the nucleus of this cell. Okay, now a lot of work was set to set the stage. In preliminaries, Song Yen found that the vesicles isolated from the non-tumor cell line, which have this Cas9 and guide RNA, can be swallowed up by the breast cancer cell line by a process called endocytosis. You can show that these vesicles actually get internalized into the reporter cell. And so now the question is, once they are internalized, will the vesicle inside the endosome into which it's been taken, will that membrane of the vesicle engage in membrane fusion, which would be required to spill its contents from the vesicle into the cytoplasm of the target cell? It's a critical feature. All right, so here are the results. At first it was very disappointing. Here are the results in the first experiments. With exosomes purified from these non-cancerous cells, we could add them in 100,000-fold excess into cells of the tumor cells that have this sensitive reporter. And even in the presence of a 10 to the 5th to 1 excess, the expression of N-luciferase in the reporter was very modest, only a 50% increase in the level of that enzyme compared to a 100-fold increase that you saw in the previous slide. And we could show, once again, that if you actually directly express Cas9 in the acceptor cell along with the guide RNA, in this case a 60-fold increase in the expression of N-luciferase. And even though it's a modest level, it was still at least somewhat dose-dependent. You could see by changing the level of vesicles, there's a modest increase. So there's something real going on, but it's really very inefficient. Indeed, it really, I was not surprised by this, because the critical thing to make an exosome functional in this kind of delivery would be the presence of some kind of protein fusogen that allows membranes to fuse. We know about these fusogens, envelope viruses, coronavirus, HIV, all the membrane envelope viruses in nature deliver their nucleic acid cores into cells by this process of internalization and membrane fusion. Viruses have evolved these fusogens, but exosomes, we think, don't have them under normal circumstances. So I wasn't surprised by this. Nonetheless, it was very disappointing, and so partly out of frustration, we did one last experiment, which was not to isolate vesicles from a donor cell, but simply to take the donor and acceptor cells and mix them together so that they had possible physical contact. Could the cells communicate Cas9 from donor to acceptor if the cells had the possibility of physical contact? And that worked really surprisingly well. Let me break down some of the data in this experiment. The experiment started with an evaluation of the donor cell line. It's called HEC293. It's a non-cancerous cell. And vesicles were isolated from these donor cells. These cells expressing Cas9 in the way that I've described were co-cultured with a variety of different tumor cell lines, including this one that I've already described, this breast cancer cell line. And they were cultured in the presence or absence of biotin, which would allow the Cas9 to remain bound to CD63 or to dissociate. And under almost any circumstances, with most but not all tumors, physical contact allowed a very substantial increase in the expression of N-luciferase. So Cas9 clearly, in this case, was being conveyed somehow between donor and acceptor cells. Now, surprisingly though, if HEC293 cells, the non-transformed donor cell line, was set up so that it could be both a donor and an acceptor, co-culture of the two forms of HEC293 cells produced no such transfer. In other words, there was some asymmetry in the ability of a non-transformed cell line to communicate in this way with a cancerous cell line. So that was really surprising and quite interesting. Now, what could this be? What is it about these kinds of cells that would allow such conveyance between cells dependent upon physical contact? Well, there is a quite pronounced literature over many years, at least in cell culture, where cells, when incubated to near confluence, send out small tubular connections, sometimes called tunneling nanotubes, that are formed from a donor cell and can touch and occasionally fuse at the site of adhesion to a target cell to create a thin cytoplasmic bridge of continuity. These are known to exist, but the means by which they form and fuse has not been explored. We know, for instance, that these tunneling nanotubes require a filament network to form, consisting of a structure-forming protein called actin. And we know from various papers in the literature that not only molecules like proteins and RNA, but organelles, mitochondria, lysosomes, protein aggregates can be conveyed between cells within these tubules. The tubules come in different sizes. Some are very thin, some are very thick. Cancer cells make them in a rather thick dimension, from 1 to 2 microns in diameter. Now, looking on the left, these tunneling nanotubes may project and adhere to a target cell, but not fuse, or they may project and then fuse. The ones that don't fuse are called closed-ended connections, and the ones that do fuse are called open-ended connections. So we wanted to explore what was going on in our circumstance. So what we did was we set up a situation where we could visually distinguish donor and acceptor cells. The donor cells in this case, the non-cancerous cells, HEC293, have green fluorescent protein being expressed in the cytoplasm. And the acceptor cells are labeled with a fluorescently tagged form of a protein lectin that binds to the cell surface and then gets internalized into the cell. So these cells can be seen as purple and the donor cells can be seen as green. And Tsongyang was very patient. He looked at lots of cells and he could see in some minority of instances that there are indeed fairly thick tubular connections that you can see forming between the donor and the acceptor. On closer inspection, in these instances, very often you could see continuity between the cytoplasm of the donor and acceptor cells and some organelles flowing quite actively from the donor cell into the acceptor cell. So this would be an open-ended tubular connection. We can even capture images of these cells by a technique called correlative light and electron microscopy, where an example shown in the upper left is such a cell with a tubular connection where the donor cell is green and the acceptor cell is purple. You can process the sample, examine it by fluorescence microscopy, and then you can fix and dehydrate and embed the specimen and cut thin sections for transmission electron microscopy. And if you mark the sample appropriately, you can identify the very cells in contact. And here is one such example of a continuity between a donor and acceptor cell where the contour of the adjoining plasma membrane has been highlighted and a breach in this connection is seen at the bottom, which must have represented a membrane fusion event that allowed continuity and could be seen as a broken plasma membrane in this junction. Now, I told you that this transfer requires some asymmetric contribution of donor and acceptor cell. And I told you that the donor cells that we've used, these non-cancerous cells, can be set up to be both a donor and acceptor, at least in principle. And I told you that in those circumstances, there is no effective transfer of Cas9. Indeed, on close inspection, we could see that in such incubations, tubular connections form nonetheless, but they remain closed-ended. There is no fusion. You can look even after a few hours and there appears to be a log jam at the point of adhesion between the donor and the acceptor, with little indication of any cytoplasmic continuity. So this is an example of a connection that does not go on to represent membrane fusion. There's something missing, some fusogen missing in this. All right, well, let me conclude with the most recent experiments that allowed us to publish the story, and that was in the discovery that although exosomes may not be very efficient in their delivery, possibly because they lack a cellular fusogen, the human genome does in fact encode a virus-like membrane fusogen. It's a molecule called syncytin, which is shown here. So viruses like HIV or coronavirus make membrane proteins that are delivered in a virus or to the cell surface. And one of these molecules from an ancient retrovirus was hijacked by the human genome maybe 10 or so million years ago. And this molecule of viral origin has now evolved to serve a purpose in early embryonic development. So in the early embryo, before implantation, the outer layer of the embryo is a cell called a trophoblast, and the trophoblast synthesizes this protein syncytin to allow the trophoblast to form a fused compartment of cells by the formation of what's called a syncytium. This syncytium of trophoblast cells gives rise to the human placenta. So it's a very unusual symbiotic relationship between an ancient retrovirus and human development. Anyway, we know a lot about this syncytin molecule. Some of the features of it being required to be active as a membrane fusogen are highlighted here. I'll skip the details to go on to tell you that we were able to show that syncytin is expressed in the cancer cell line. Indeed, it's known in the literature that human tumors upregulate the expression of syncytin, whereas most tissues in our body do not express this protein. Human tumors do, and it may be to exploit some function that allows the tumor to survive. So let me cut to the chase and show you here that if you block the production of syncytin in the tumor cell line and you set up the assay as I've described, you fail to see, you can see tubular connections that form, but they do not go on to promote membrane fusion. It's a log jam. They adhere, but they don't fuse. So we can conclude, finally, that in our assay, the contact that forms between donor and acceptor cells depends upon the presence of syncytin in the acceptor cell, and in other experiments depends upon a receptor on the membrane of the donor cell. These produce a continuity. Oops. We go back to my presentation. Oops. No, no, wait. Okay, so we can show that in these experiments, the tubular connection forms, membrane fusion occurs, and cytoplasmic continuity allows Cas9 to be transmitted into the cytoplasm in the nucleus of the target cell. Now, there are many implications of this, in particular that relate to tumors and how they survive, and I'd be happy to discuss this with you. But I go back to the original question. The exosomes were not functional, I argue, because they lack a fusogen. However, there may be avenues of communication in our body that exploit the presence of syncytin in certain tissues that will permit exosomes to be functional. Indeed, it's possible to consider engineering the expression of syncytin to produce exosomes that are capable of delivering content. And this is a very attractive possibility to use exosomes as a therapeutic delivery vehicle, relying on the fact that there is an endogenous fusogen that will be immunologically tolerated and could then be used to produce an exosome that could be very useful in biotechnological applications. Many things to discuss. This is the research group. I'm going to highlight the contributions of this brilliant postdoctoral fellow in the middle there, Sonia Zhang, who's done all the experiments that I've described. Thank you.
For this excellent presentation of a very exciting subject. I hope that the results will be very positive for the cancer treatment in the future, and I wish you the best continuation of your job.
Thank you.
Now I will pass the chair to Professor Adelu to continue.
Thank you very much. So, briefly to introduce myself, Rainer Adlung from Kiel University. We have time for a lot of questions, if possible, if you would be so kind.
Oh, is there time? I thought maybe I'd
it's super exciting. Yeah, young Chisinau made this very good move to start half an hour earlier.
Okay.
So we are still in schedule, and it would be really, yeah, thank you.
Thank you very much. You have shown avenues for the future biomedicine. Very specific question. How many exosomes do you need in order to perform your identification? Can you scale down this number? Because in the spirit of nanomedicine, one should work with simply single exosome. I understand that this is for the future, but what is your vision?
So there are two different avenues where exosomes could be useful. The first, which is amenable to very small scale, as you suggest, is in diagnostics. So I can tell you later. You can look online. There's a paper that I'm an author on that's on the bioarchive that you can look at. But there are very clever ways of using just small samples of human blood plasma to detect the presence of multiple tumor antigens on individual part vesicles, but measuring this in a population of millions of such vesicles. There are millions of them in just a few microliters of blood plasma. And there's a way that I can describe to you that a company in Boston has devised to monitor the coincidence of 3 different tumor antigens on individual particles in populations of particles. And they're, as a result of this, they've been able to have a much more sensitive detection of tumor antigens represented in ovarian cancer that is much more sensitive than the current diagnostic technique. And so they can actually pick out patients who are at stage 1 who have no evident pathology other than this expression of tumor antigens. So for diagnostic purposes, looking at small populations will certainly be feasible. Now, for therapeutic applications, you need more material. The problem with exosomes is that cells don't manufacture enough of them to scale up to treat lots of patients. So that's going to be a logjam. I think more likely it would be possible to engineer the production of synthetic vesicles. Similar to, but perhaps more elaborate than the lipid nanoparticles that are used as antigens, sources of antigens for now mRNA therapy. In fact, there's another company that I consult for again in Boston that has found that formulations of lipids from plant and microbial origins produce vesicles that can encapsulate messenger RNA and deliver them very efficiently to different target tissues of the body. So it's a very promising approach. They don't understand how this works, but it does work. This can be scaled up to an enormous quantity, not relying on cells at all. So I'm very hopeful that both of these goals can be achieved in the coming years.
Thank you.
Sure. Okay. Yeah. Next question, please. So maybe I can place a brief question. So forgive me, I'm a material scientist, But I strongly believe, I think it's a theme of the conference, that we all should work interdisciplinary together. So what I learned, I found quite interesting about these nanotubules and these communication channels. So I have the feeling that there's still some improvement needed for the in vitro cultures in order to really have the kind of right model. What is your opinion about that? Because maybe you can ask some material science guys to design something.
Yeah. Well, I view this as a means of detecting some natural phenomenon that probably occurs in the body and not just in cell culture. You can imagine that in a tissue like the brain, which has billions of cells, detecting the presence of these tubular connections between nerve cells that are juxtaposed in close proximity is very difficult. Nonetheless, there are some examples in the literature where you can see these kinds of tubular connections forming, and the question is, what role do they normally serve? So that's a kind of a biological question that I think can be addressed. What intrigues me now is our observation that this molecule syncytin is upregulated in tumors, and so there's one paper in the literature that has shown that if you take a tumor in the form of a xenograft and you introduce it into, I forget which organ, in a mouse animal, you can show that the tumor cells project tubules to touch and then fuse with non-transformed cells at the margin of the tumor, and in doing so they hijack mitochondria from the target cells back into the tumor cells. So if that's true in tumors, in solid tumors, this may be a means by which tumors nourish themselves, and if you could block that by immunologically blocking the function of syncytin, you might starve a tumor cell. So that's another kind of immunotherapy that could be considered. I don't know if anybody's doing it. It's an idea that's out there now, so it's a really interesting possibility.
Fantastic strategies. Yeah, so are there more questions? If this is not the case, then let's thank the speaker again. So the next speaker will be Professor Testemitanu, who I had the pleasure to meet, I think 14 years ago, something like that, at the first workshops here in Moldova. And yeah, as I already said in my last question, I really like this interdisciplinary approach, and we have nanotechnology and biomedical engineering at the same time, and I got to know over all that time Professor Testemitanu as the expert for gallium nitride, and it was further and further developing during the years the applications of gallium nitride, so from light-emitting diodes, which are now everywhere present, now to also power electronics. But you showed also some biomedical applications, so we are very much looking forward to your talk.
Thank you very much, Professor Adelu, and I take this opportunity to thank Professor Andy Shepman for the really very interesting presentation. So I will talk about gallium nitride. Actually, it's a material that plays a major role in the development of micro nanoelectronics. We represent the National Center for Material Standard Testing, Technical University of Moldova, and the Academy of Sciences of Moldova. We deal with gallium nitride for many years, and well, here you see the content of my presentation, and also I will underline that taking into account the very important properties like of gallium nitride, like direct and wide band gap, that it has chemical stability, stable at high temperatures, lighting technologies, blue light emitters are based on gallium nitride and alloys. And it's a perfect material for high power, high frequency applications. And it's biocompatible, and actually we demonstrated this in a series of experiments. You see here that gallium nitride is a leader in, if we speak about high frequencies and power applications, because we have indium phosphide very good for higher frequencies, but not good for higher power. or we have silicon carbide, very good for higher power applications, not good for higher frequencies. Gallium nitride is good for both. And another very important issue is that gallium nitride with its solid solutions cover a very large spectral region. It's a unique material system. You can compare with other material systems like you have with gallium arsenide, gallium phosphide, and so on. And you see that the band gap of materials in the system, gallium nitride, aluminum nitride, lithium nitride, it's really fantastic. So these materials are very close. Gallium nitride, zinc oxide, and we took this into consideration when planning some experiments. And you see that actually both materials have higher binding energy of excitons. In zinc oxide, it's 60 millielectron volts, 2.5 higher than thermal energy. And the most important for us is that the mismatch between gallium nitride and zinc oxide is 1.4%, is negligible, and that's unique. And with the temperature, this mismatch doesn't change, and this is also very important. Gallium nitride has rapidly transformed the world by enabling energy-efficient white light emitting diodes and promising energy-efficient power electronic devices. And I would like to say that in our mobile phones there are a lot of devices, nano devices based on gallium nitride. So we are in permanent touch with one of the world leaders in gallium nitride, with Professor Hiroshi Amara. He got, along with Professor Isamu Akasaki and Professor Shuzo Nakamura, the Nobel Prize in 2014 for the invention of efficient blue light emitting diodes, which has enabled bright and energy-saving white light sources. I would like to know that Professor Hiroshi Amara is a honorary member of the Academy of Sciences of Moldova. He developed, he delivered many presentations at the Academy of Sciences of Moldova. The last one was in June 2023, and it was a presentation, a report for young students, for pupils from lyceums. There was a summer school. What are the most important characteristics of a scientist? I would like to know that it was really a very interesting presentation, and our goal actually was to attract young people to science. But the titles of other presentation you can see here on this slide. So, gallium nitride, zinc oxide, three-dimensional micro-nano architectures. We started this activity about probably 10 years ago after a talk with Professor Rainer Anderson. It was in Grenoble, probably, about 10 years ago. It was an idea, a crazy idea at the time, but with the time we demonstrated that it works, and we just took into account that the mismatch between gallium nitride and zinc oxide is negligible. From general points of view, the epitechs should work. And it worked, and we deposited gallium nitride on the zinc oxide, the tetrapods available at the University of Kiev. And the interesting issue is that during the deposition, the zinc oxide is removed. And as a result, we got hollow microtetrapods. That means with very thin walls, and actually 10-20 nanometers could be a little larger, 10-20 nanometers the thickness of walls, but the wall actually is not completely consisting of gallium nitride. There is an outer thin layer of zinc oxide on inner walls. The zinc oxide survives. We tried to remove completely, but it's not possible. That was also a very, very interesting observation. The first paper was in 2019, and afterwards we published a series of other papers on different phenomena. But of course, this came as a result of international collaboration, and actually what we discovered as a result of this, the duality of properties. That means this material, we called it iron light, so is due to the fact that each hollow microtetrapod so has lateral walls hydrophobic, while the free ends of arcs are hydrophilic. Why? Because the free ends of arcs represent polar surfaces, and the polar surface, charged surface, it attracts water because water is dipole. And what we actually claimed in that paper, that we got a new material, quite new material, that exhibits two properties at the same time, hydrophobicity and hydrophilicity. And you can see here hydrophobicity obvious. So this is the real picture. That means the shape of the drop on the material shows that it's a hydrophobic wetting. While when you try to take out the material from the water, water doesn't allow you to do this, as you see in the lower part of to the right. That means at the same time we have hydrophilicity and hydrophobicity. You take the material, press against water, water runs away. You would like to take the material from the water surface, water doesn't allow you. You will see the formation of a column, water column. And you see here pictures that shows the formation of a water column. That means demonstration of hydrophilic wetting. You can see here the formation of the column. This is a charged stick, and it attracts the material. So slower, you will see the formation of the water column. Yeah. Okay, what we observed that this artificial insects, we call them artificial insects, hollow microtetrapods, they interact on the water surface and actually form a membrane. And it's similar to the interaction of fire ants during the floods, because they interact, form live raft, and survive as a result. And you can see here just one microtetrapod on the water surface. We made experiments with a huge number of tetrapods and succeeded to create membranes, floating membranes, and you can see to the right here that we can also load the membrane with the color drop, and the membrane will continue to float. And so we decided to study these membranes, artificial membranes consisting of interacting hollow tetrapods. I remind you that the thickness of the walls of these hollow microtetrapods is just a few tens of nanometers. So here you see the direct aphoretic actuation of floating graft. That means the stick is charged, negative charge. The same, of course, in case you will have a floating graft with a drop of liquid on it. And now I will show you unique results related to the flexibility, hardness, and self-healing properties of iron organometallic membranes. So you will see now, the membrane was created on one arm of communicating vessels. That means in another arm we remove part of the liquid, or we add liquid. And as a result, you see that we have this kind of oscillations. And the membrane was prepared in equilibrium when we had a flat surface. That means you see that the membrane is actually flexible, and in that case you see many vibrations. Here just one increase of the water in the other arm, and you see how it behaves. Fantastic that we demonstrated the flexibility also with marbles, because when you cover a drop of liquid with this kind of artificial insects in equilibrium, and afterwards you just add some liquid over the membrane, of course, enlarges its surface, or we make it vice versa. We allow some liquid to evaporate, and of course the penetration of tetrapods increased, and in this case we got consolidated liquid marbles. I will tell a little later. So you see such a membrane, just to estimate the of course, there is a threshold. After you reach the threshold, cracks will appear. The interesting issue is that when you go back, cracks disappear, and the next experiment, the cracks may be in another place. So that's really fantastic. I mean, it's artificial membrane consisting of microtetrapods, dimensions tens of micrometers. The thickness of the walls is 10-20 nanometers, and you have such kind of behavior which looks really very, very impressive. I remind you that we just add water to another arm of communicating vessels to understand why this happens. Yes. Okay. Well, here you see the electrophoretic actuation of inorganic nitride. Actually, very interesting, and I'm quite sure that this phenomenon can be used in nanomechanics, nanorobotics, and so on. We simply have no relevant infrastructure for this, but probably we can study by joint efforts. And to the right, you will just evaluate the elasticity of arm of tetrapod, hollow tetrapod. You see how so the tetrapod is deformed. Here the tip of the atomic force microscope just forces the tetrapod to change its shape. Okay, and I told about liquid marbles. What is a liquid marble? It's a drop covered by nanoparticles, by definition. In this case, the role of particles is played by hollow tetrapods. And you see in cross-section, schematically, you see one actually of the first liquid marbles that we created, and picture how it floats on the surface of the water. And I told you about consolidated liquid marbles. That means a marble that is covered by this kind of particles. And when you remove a part of it, simply part of the liquid evaporates, then the interpenetration between microtetrapods increases, and you get a consolidated liquid marbles. It means that, for instance, here between two pieces of glass, and you deform the liquid marble as you like, but it survives. And it survives also on the surface of the water, subjected to intense ultrasonic treatment. And here, well, you see liquid marble actuation by electric field. It's a charged stick. That means you can control by electric field the floating of the marble. Self-propelled liquid marble is based on aerogallium nitride. We added some alcohol, volatile compounds to the liquid droplet, to the water droplet, and as a result we got marbles that show rectilinear movement, or rotation of the surface, water surface. You see rotation actually at a very high speed. We investigated this and we found that actually the higher speed of rectilinear movement of rotation is related to the fact that the marble is skating on the surface water. And I will show you later. That means you see here the speed of rotation. It's 12.5 rotations per second. At the beginning, it means 750 rotations per minute. So you see how to the right schematic picture, how the three ends of the tetrapods that covers the liquid, they are tethered to the surface of the water, trapped to the surface of the water. And when you have a gradient of surface tension, the marble will simply skiing on the surface water. And what? Yeah, you will see that nature already invented so insects. Their legs are hydrophobic, while the claws are hydrophilic. And it flies and at the same time skiing along the surface water. It's the same in our case because we have the arms of the tetrapods hydrophobic, while the three ends hydrophilic. And as a result, we have the marble skiing on the surface of the water. Here we observed a quite fascinating phenomenon, so liquid marbles rotating in pulses. It was not easy to find an explanation, but we succeeded, and I will draw your attention that in fact we observed that the marble intends to take off the surface of the water. And the idea, the concept that can explain is this following. Some arms of tetrapods play the role of helicopter vanes. That means when the speed of rotation reaches threshold speed, the marble tries to take off, but at the same time, thousands of nano currents of water appears, and of course it will not have the possibility to rotate, and it falls on the water surface. And you can see here the speed of rotation as a function of the weight of the marble, and obviously we have a threshold speed. The experiment shows that we really have a threshold speed, and the threshold speed shows that probably the explanation that we proposed is feasible. And pendulum-like oscillations is another very fascinating observation. That means you see the experiment, liquid marble at the bottom with the glass vessel. You can see schematically on the top left. So marble shows pendulum-like oscillations to the right, to the left. We observed this for various marbles. You can see here just oscillations by half of period, let's say. And at least in this case it's easier to explain because actually this self-propulsion is related to a gradient of self-extension. And imagine that we have self-extension, we have a high evaporation here, low evaporation here. That means there will be the gradient of the self-extension and the marble will move, for instance, in that direction. When you return 180 degrees, of course, these 2 points change and it will go in the gradient of self-extension back. At least here it is possible to explain, but we are doing more experiments to understand this. Biomedical applications of gallium nitride nanoparticles and iron gallium nitride. Well, we succeeded in forming a protein corona on zinc oxide doped by aluminum, and we tried also on gallium nitride. This was under the European grant, and here we have a collaboration with the partners from that project, Hannover Medical School and joint Center of the European Commission. Actually, as a result, what we observed here that we can actually cover the zinc oxide particles with, how do you mean, and it is not possible to have a protein corona on gallium nitride. This actually showed that we made use of this difference in the behavior of surfaces of zinc oxide and gallium nitride, because we are able to create different structures having the interface between these two materials. So the paper has been published in Journal of Applied Biomaterials and Functional Materials. Just to show you, in case of tetrapods, we have the interface between gallium nitride and zinc oxide. We are able also to have zinc oxide on the inner surface, on both surfaces, inner and outer surface, and so on. It means that on zinc oxide we can actually create a layer of albumin proteins. So gallium nitride zinc oxide hollow nanoparticles for biomedical applications. Actually, the process was the same as in case of tetrapods. We started with zinc oxide nanoparticles, deposited gallium nitride. We got gallium nitride hollow nanoparticles. We found that these nanoparticles interact with living cells, and actually living cells attract these nanoparticles. Here we demonstrated that actually gallium nitride is biocompatible, because if we compare the impact of the zinc oxide and gallium nitride, that zinc oxide at relatively higher densities, concentration, just kill the cells, while gallium nitride not. And here you can see the behavior of cells to the left without nanoparticles, to the right with nanoparticles. The cells were found to collect the nanoparticles surrounding them. Nevertheless, the mobility and the proliferation activity of endothelial cells are not affected by the presence of gallium nitride nanoparticles in the medium. What we demonstrated in these experiments that gallium nitride hollow nanoparticles penetrate into the cells, and we found them in vesicles inside the cells. And we doped the gallium nitride with iron. That means we just have, in this case, nanoparticles with magnetic properties. And it was found possible to demonstrate that marked cells, so we can influence upon their behavior using a magnetic field. And you can see here the distribution of the cells in case there was no magnetic field, and the distribution of the cells when we have, you can see here fluorescence pictures of endothelial cells targeted with gallium nitride diamond nanoparticles and incubated in control groups and in magnetic field gradients. That means the lower pictures was under the influence of magnetic field gradients. After a few hours of incubation, a non-uniform distribution of cells exposed to magnetic field could be notified, while cells targeted with nanoparticles and incubated in normal conditions without magnetic field are distributed uniformly on the surface of the culture plate. I will show these results that actually we got some years ago, but probably there could be a continuation, because having these hollow nanoparticles inside with magnetic properties, and also gallium nitride is also piezoelectric material, could open possibilities to have an influence, remote influence on the behavior of the cells. Of course, on cells marked with nanoparticles. So just will mention that we demonstrated that iron gallium nitride is a shielding material for both gigahertz and terahertz electromagnetic fields. Here we have for this experiments in collaboration with the Institute of Microtechnology in Bucharest. The same for terahertz, and shielding effectiveness of iron gallium nitride exceeds 40 decibels, being among the best terahertz shields known today. The value of 40 decibel is required for industrial application and is fulfilled in the frequency band of 1.12 terahertz. So the interesting issue is that the iron gallium nitride is actually more rare than material. It's when we have hollow tetrapods, it means the degree of porosity maybe 97%, 98%. That means, and it shields better the electromagnetic fields than other materials. There are materials based on metals and so on. So the last demonstration, fluorescent microangios. So this also was very, how to say, good collaboration with the German researchers from Kiel, from Dresden, and we actually, you see here that under illumination we have movement, and it is possible also to demonstrate some other properties of. You have here one larger and another smaller microangio. So just will mention that this is the 6th edition of the conference. We started in 2011. So I would like just to know that we identified some persons that attended practically all the editions, and one person really all the editions attended, and we decided is Professor Schotter here? Not here. So we decided to give special award to Professor Fonin for his attendance of all the editions, and well, we will then give the conference give the award, and also award to to Dr. Sergio Relian, because he was responsible for the preparation of materials. Of course, the whole organizing committee with Professor Schonda, but he played a very important role, and he also will get an award. Okay, conclusions. They are here. I will not repeat. Thank you very much for your kind attention.
Yeah, thank you very much. Coming all the way from, I would say, serious semiconductors to biomedical engineering. Especially, I think it's fascinating that you have like microscopic or almost macroscopic models like of cells, like these liquid marbles. So one get new ideas, maybe to study behavior and treat that. Yeah, well, the talk is open for discussion. Who has questions?
They are watching.
So, Professor,
Yeah, please.
maybe I can ask a very special, very specific question. At the beginning of your talk, you talked about the lattice mismatch between gallium nitride and zinc oxide. And in what sense that lattice mismatch is just in a 2D interface? Or
2D interface, yes. Because this is the same structure, hexagonal structure. And in 2D we have the lattice mismatch over 1.8%. And another interesting issue is that this mismatch does not change with increasing the temperature. And that's very important, otherwise we create devices that have higher temperatures. And this is really a unique situation. Dear Professor Schonda, please come. Give the listen. This is to Sergio Relian, Nicolae of the Academy of Sciences of Moldova, for his contribution to the organization of the conference, starting from 2011 and until now, this 3 years. Everything okay? So, congratulations, and this is also yours. This is for me. Professor Pomir, medalja Dignity Continuity, pentru succese remarcabile for fascinating achievements in theory and development of theoretical physics, and for contribution to the conference, he attended all 6 editions. Congratulations. Okay. Thank you very much. Very nice. Look. Thank you very much.
Yeah, so after this spontaneous emission of awards, we're coming now to the last plenary session. I'm very happy to announce Professor Mimura, who is, I would say also long-standing contributor to this series of conference, and it's always a great pleasure to see his progress. And here again, we can see this in the title. Microelectronics are used, electrical devices in order to help medicine. So we are looking forward to your talk.
Thank you, Chairman. So thank you, organizing committee, for inviting me to this conference. I'm Hideno member at Research Institute of Electronics, Shizuoka University. So I'm a former director of the Research Institute of Electronics. Now it's Professor Oki is the vice director of the Research Institute of Electronics, and Professor Kimura is the new director of the Research Institute of Electronics. So both directors attended this conference. So our institute, Research Institute of Electronics, is the birthplace of the all-electronic television system. So the late Professor Kenjiro Takayanagi discovered, invented the television system. So this is our institute. Okay, so the Professor Takayanagi displayed the Japanese character. E is in alphabet is A. So the displayed in the brown tube in 1926. So since then, our institute is designated as the center of excellence in the imaging devices. So our institute is developing several imaging devices. So recently, our institute developed the 8K, so super high vision image sensor with the Japan Broadcasting Association. And then, we offered such a 8K super high vision image sensor to the medical, the researchers. So this is the outline of my talk. So today, I'll talk about the rehabilitation equipment. So using the nanomaterials. So our institute is very strong on the image, strong on the sensing devices. And recently, we collaborate with the medical researchers and offer the sensing devices to the medical researchers. And then I'll talk about the carbon nanotube. So we have developed the carbon nanotube storage sensor, and then our group has developed the data globe using the carbon nanotube storage sensor for finger motion detection. So I'll talk about these devices. And then we have applied the data globe to the rehabilitation equipment for moving the paralyzed fingers. So that I'll talk about this rehabilitation equipment. So that's its conclusions. So the carbon nanotube is a very popular material. So the carbon tube has a cylindrical nanostructure of the graphene sheet. So in Japan, Dr. Iijima discovered the carbon nanotube in 1991. So our deposition method is a very simple one. So we put the quartz substrate and iron chloride catalyst inside the furnace. And then furnace is evacuated at the pressure less than 0.1 Pascal, and we heat the 820 centigrade while keeping the vacuum. And we flow acetylene gas. This is very simple method. So the ultra-low multi-wall carbon nanotube grows on the quartz substrate. And our growth is occurred at the bottom of the carbon nanotube. Our method is a rapid growth, so that typically 21 millimeter carbon nanotube is grown for the 20 minutes. And so this is carbon nanotube. So we have built up the startup company. The name is Hamamatsu Carbonics Corporation. This company produces cellular products such as carbon nanotube tape and carbon nanotube yarns and carbon nanotube array and carbon nanotube sheet. So these products are 100% carbon nanotube. And then Hamamatsu Carbonics Corporation, so the cell, so that these the products. Okay, so we have developed the carbon nanotube strength sensors. This slide shows a cross-sectional view of the carbon nanotube strength sensors. So we put the aligned carbon nanotube tape on the polycarbonate flatten regime PCU substrate, and then we adhere the aligned carbon nanotube tape with the path electrode and the conductive base. The aligned direction of the carbon nanotube is parallel to the strain axis. So this slide shows the cyclic strain resistance characteristic of the carbon nanotube strain sensors. This axis is resistance, this axis is strain. So the resistance is linearly proportional to the applied strain. So the lower strain, so lower resistance. The higher strain, the higher resistance. So this slide shows the dynamic strain resistance characteristic of the carbon nanotube strain sensors at a frequency of 29 Hz. So the blue curve shows the applied strain, so red curve shows the resistance. So resistance change, so this is the resistance, this is the resistance. Resistance change follows the temporal change of the applied strain, so very well. So until the frequency of 30 Hz, the sensor responds so very fast. So we have developed the data group to detect the fine finger motions. So these are the carbon nanotube strain sensors. The carbon nanotube strain sensors so detect the angle of the fingers. So this data group so collects the data of the fine finger motion from the angle of the fingers. Okay, so using the carbon nanotube, so we have developed the rehabilitation system. So this is because there are many stroke or seminal injury patients suffering from the after effect of the disease. So such that patient cannot move so their fingers of the paralyzed hand on his own. However, when the proper electric stimulation is applied to the paralyzed hand, the fingers of the paralyzed left hand can be automatically moved. So the muscle so respond the electric stimulation. Okay, so this function, so we call the functional electric stimulation. So that we apply the electric pulse to the paralyzed left hand. So the fingers, so the automatically so the move. So that is when the proper electric pulse is applied to the paralyzed left hand through the electrode, the fingers of the paralyzed left hand can be automatically moved even though the patient cannot move his fingers of the paralyzed left hand on his own. So using this functional electric stimulation, we have constructed the rehabilitation system. So the rehabilitation procedure is so as follows. In the step 1, so we put the data flow on the both hand. So this hand is a normal right hand. So the patient can move his fingers. So for this hand. So this hand is a paralyzed left hand. So the patient cannot move his finger. So in this paralyzed left hand. This is an electrode. So we apply the electric pulse to this electrode. So the fingers, so the automatically move. Okay, and so in step 1, if the patient makes each finger's posture from 1 to 5, which is normal, so right hand. In the posture 1, the thumb is bent. In the posture 2, the index finger bent. In posture 3, so the middle finger bent. In posture 4, is the ring finger bent. In posture 5, so little finger bent. So the right data node, so detect the data of each finger motion, so like this, of the normal right hand and register the data of the each finger posture on the PC. And when the posture is 1, its data is R1. In the posture is 2, in this case, data is R2. The 3 is data is R3. And so electrode, so the multi-electrode consisting of 4 times 6 electrodes are placed on the inner side of the forearm for the paralyzed left hand. So the PC selected 1 active and 1 return electrode out of 24 electrodes. So there are 552 for 24 electrodes. 24 times 23 is 552. Combination to choose the pair of the active and return electrodes. Okay, in step 2, the pain current pulse is applied to the active electrode. The finger of the paralyzed left hand is automatically moved. So the left data below detected the data of each finger of the paralyzed left hand and registered the data of each finger on the PC. So we choose the 1 active electrode and 1 return electrode. So the 550, we get the 552 data. So 550 data from the L1 to L552 are registered on the PC. So we comparing the R1 to R5 registered by the right hand. So with the L1 to L552 registered to the left hand. The left combination of the active electrode and the return electrode is selected for each posture of 1, 2, 3, 4, 5. For example, the posture 1, so this combination is selected. So this is the active electrode, this is the return electrode. The posture 2, this combination is selected. This is the active electrode, this is the return electrode. In the posture 3, this combination is selected. In the posture 4, this combination is selected. In posture 5, this combination is selected. So in the step 3, for example, when the normal right hand makes a posture 1 on his own, in this case, so when we current pulses are applied to the active electrode according to the combination selected for the posture 1. In the posture 1, So we selected this combination is the best to make the posture one. So in the posture one, this right hand so makes the posture one, so that we apply the pulse for this using this combination. So in this case, so paralyzed left hand automatically makes the posture one. So that at the moment, the patient also try to bend his thumb of the paralyzed left hand on his own so strongly. This is important. So the when we apply the electric pulse, this the muscle so automatically responds to the electric pulse, and this finger makes the posture one. However, the patient so strongly try to bend his thumb of the thumb finger on his own. So it's a rehabilitation. In the posture 5, so similarly, when the normal right hand makes the posture 5, current pulse is applied to the active electrode according to the combination selected for the posture 5. The posture 5 is this selection, this combination is selected, so that we have to apply electric pulse according to the this combination. So then the paralyzed left hand automatically makes the posture 5. So at the moment, the patient tries to bend his little finger of the paralyzed left hand on his own strongly. The first patient cannot bend his little hand, but he tried again and again so that he can move, he can he can bend his little fingers. Okay, this is the example of the rehabilitation. So first, so normal hand makes the posture. So 1, 2, 3, 4, 5. So when the current pulse is applied to the active electrode on the paralyzed left hand, the paralyzed left hand so makes the same posture. Paralyzed left hand makes the same posture as made by the normal right hand. Okay, this is the posture 1. So the thumb bend. So this is posture 2. It's index fingers bend. In the posture 3, in the mistake. It's a middle fingers, so the bend. In the posture 4 is ring finger bend. In the posture 5, the little finger bend. So the paralyzed left hand so makes the same posture as made by the normal right hand. So that these are the rehabilitation of the paralyzed left hand. Okay, so initially the finger of the paralyzed left hand move automatically only. So this is a muscle response. It's electric pulse. But so when the pulse is applied to, however, but this rehabilitation so continues, the patient will be able to move his fingers on his hand on his own. So afterward. Okay, let me conclude my talk. So we have developed the carbon nanotube strength sensors. So using carbon nanotube strength sensor, we have developed the data globe for finger motion detection. So we have developed the rehabilitation system that combines the functional electric stimulation and the data globe for moving the paralyzed fingers. So we have demonstrated the system. Here the paralyzed left hand makes the same posture as made by the normal right hand. So using this method, so patient can train multiple motions using the functional electric stimulation. So thank you very much for your kind attention.
thank you very much for the interesting talk. Are there questions?
First of all, great presentation. I think whenever you apply a signal, it's a response from the muscle. So that's programmed into your experiments, I suppose.
So that I make some electric pulse and apply to the electrode. So the muscle, so response, is a pulse of the electric pulse.
Right.
So, but this electric pulse we have to allocate the best pulse shape, best shape of the electric pulse.
Thank you.
Well, thank you for your really excellent presentation. And I would like to ask a question. Well, this you developed for real fingers, but imagine that we have a terrorist and we should stop his actions suddenly because he has emotion and actually detectors, sensors can detect this. And having such kind of, just stop him at the proper time.
If we don't apply this on the electric pulse, it's the finger cannot make the soda.
I see. I see. Yeah. Professor Hidenori Nomura is also a permanent attendee of this conference, like Professor Adelung, and we thank you. And he is also a honorary member of the Academy of Sciences of Moldova.
Yeah, further questions? Yes, please.
Thank you very much for the very nice presentation, and I hope that this will be proven that in the future that rehabilitation could, through rehabilitation, the hand, the paralyzed hand could learn. But you mentioned that, but you didn't went a little bit further. Are there examples of paralyzed hands that have been able to, let's say, educated in how to deal alone?
Yeah, so this rehabilitation system now started, so we discussed with a doctor, so medical doctor, and so that it is really useful for training the patient. Yeah, but now, so this rehabilitation system is so that some doctors employ this rehabilitation system, but not popular, not yet.
Yeah. Are there further questions? If it is not the case, then thank you again.
Thank you very much.
And then I'd like to announce that, in the morning session, Professor Kalikaratis will now continue to talk after the coffee break about the healthcare professional and medical technology. And finally, you're all mad at me because I have the last talk before lunch, and I will talk about the translation of, I would say, a semiconductor drug now into pharmacy for stopping herpes viruses. Okay, I don't know how the organizers feel, but I think we are nicely ahead of schedule. Shall we meet at 12:15 instead of 12:30? If we all agree on that, then we have even a longer coffee break and later on some time will be on.
Da, okay, I will introduce Nicolae Polikarakis, and after that... Ladies and gentlemen, please take your seats. So we start the session. The plenary report number 4. The importance of training healthcare professionals in medical technology. The report will be presented by Professor Nicolae Polikarakis. I already mentioned that Professor Polikarakis plays an important role in the organization of the conference, and he is Doctor honoris causa of the Technical University of Moldova, and he is a very good friend of the Republic of Moldova, so you hope that please.
Thank you very much. I repeat it, I'm very glad to be here, and it is probably the 4th time that I'm participating in this conference, and I would be glad to participate again if I have the chance. I'm afraid I'm going to change the subject in a more practical, let's say, issue that relates to the clinical engineering work in the hospitals more than really exciting research subjects that we listen this morning. So my subject deals with how we should train users of medical devices and why we should train users of devices in the hospital environment. I'm coming from the University of Patras, but now I'm emeritus professor, and I'm as research, my field is imaging, CT imaging, computer tomography, and so on. But in the Institute of Biomedical Technology, that has been created in 1991, so more than 30 years now, we have as a goal to support Greek hospitals and probably hospitals in other countries in the future to better deal with medical devices, in management of medical devices, in safety issues, in vigilance, and so on. So we provide services to hospital and healthcare authorities with correct inventory and nomenclature, use of correct nomenclature for medical equipment, biomedical technology management through a web-based system, the web praxis, vigilance and safety, that we are also collaborating with WHO in various subjects, consulting, investment planning, etc. And we have very good relationships with WHO, the European Alliance of Medical and Biological Engineering, Global Clinical Engineering, which is a new alliance, and of course IFMBE. Now, let me give you some numbers before I start. We are speaking about medical technology, and the expansion of medical technology during the last decade is really fabulous. It's something extraordinary. We have together today the European market, that it is a 150 billion market only in Europe, and it is just almost 1/3 of the global market, which is more than 500 billion euros per year. And what is important in Europe is that we have medical technology that 95% of them are startups and small and medium enterprises. And even more important is that innovation is very active in the biomedical technology sector. Actually, in Europe, we have about 15,000 patent applications at the European Patent Office every year, and it is the first area, the first, let's say, manufacturing domain that it is far more than pharmaceutical and other domains. And so we are leading, in Europe, we are leading the innovation in medical technology. Just to give you some other numbers that probably are important, it is that we have about 12,000 groups that we arrange medical devices. We have about 1 million medical devices available today in the world market, and we have more or less 100 to 200,000 manufacturers worldwide, including all the custom-made devices, etc. Of course, some of them are very well known, some others are practiced locally, but there are thousands of medical device manufacturers. And sometimes we don't realize, but when I was very young, children, and I have been operated, I still remember in the 60s that the nurse was coming with a glass syringe, and I was afraid a little bit of the glass syringe, and the whole hospital probably had something like 100, 200 syringes. They were reused and reused. And what happened today? We have more or less 20 billion syringes and 20 billion injections that are made per year. So every day, thousands of syringes are used in the hospital. We have also, just something more complicated, 1 million of cardiac pacemaker implanted every year worldwide. In the life cycle of a medical device or a medical technology, we have a first loop of R&D. And everything starts from new discoveries and new application of new knowledge that comes out of research, like the 3 presentations they made today. But after that, there is a cycle, a second cycle that starts saying that we are going to have preclinical testing of its technology, and if preclinical testing in animals, for instance, are successful, then we are speaking about an innovation. But in order to bring an innovation, a new innovative application in the real practice, you need to make clinical studies. If it's a new technology, you need to make clinical studies and prove that this new technology is better than the alternatives that are existing so far. And then in clinical studies that are quite well-controlled studies, and probably you have an idea of what happened with vaccines, for instance, the clinical studies in the vaccines under the COVID-19 pandemic. You remember how fast they went, but this is exceptional, that they went so fast. Usually it takes time, it takes years to get a new, I repeat, new idea, a new innovation to the medical practice. And then, if this is successful, the manufacturers, they can get license to bring this material, to bring this product on the market. And to get this product on the market, you need this product to fulfill the requirements of regulations, fulfill the requirements that are related to standards, directives, and then you are allowed as a manufacturer to bring a product on the market. Once the product is on the market, there are 2 things that they are going on. One is the application of the product, and the other one is the health technology assessment. The application of the product, if it's a medical equipment, then it enters in another circle. And there, clinical engineers are important in the hospital environment to better manage this equipment. And better manage this equipment, I mean that they need to follow the equipment from the point of view of its maintenance, its quality control, safety issues, vigilance, and so on. And this is the goal, and this is the main, let's say, subject of clinical engineers in the hospital environment. So it's the correct application of medical devices. And when this technology starts to give reliable results from its use, the new technology, then health technology assessment comes, and this is a much more complicated assessment of the technology than simply, let's say, technical assessment. It's an assessment that goes further to, let's say, financial and the community, and what is the benefits of this new technology for general, the patients and the community in general. And this is the basis sometimes for reimbursement of the technology that makes this technology, let's say, successful in order to be used. And of course, if a new technology comes, the technology becomes obsolescence. And of course, sometimes, of course, every year there are products or equipment in the hospitals that they become obsolescence. So, in about more than 20 years, in 1999, a report in the United States that made a lot of, let's say, sense. It was that this report was estimating that about 100,000 deaths per year in the United States were used to medical errors. This was a shock for the medical community and also for the patients. And similar reports came out in Australia, and similar reports came in European Union. And the European Union, this is a slide from the European Union site, but it is about 15 years old, and now it disappeared. At that time, in any case, it was estimated something similar, 8 to 12% of patients that enter into a hospital suffer from adverse events. And adverse event means harmful event for the patient due to different causes. The more important is that potentially most of these events are preventable. The more important, let's say, medical errors are related to infections, are related to medication errors, are related to diagnostic errors, and to outpatient safety. And most of them are due to use errors. And this is very important to understand that the technology becomes complicated, it is used in a massive way, and so errors could happen at any moment of the patient that is in a hospital. So we have to take care and be able to better treat or to better face these problems of misuse or overuse of the medical technology. Of course, health technology, health information technology is able to assist to that, and it is proven now that concerning medication, diagnostic error, patient identification errors can be avoided by the use of information technology. However, there are some other problems that are related to the use of health technology, and it is important to use in a very correct way as well. Now, to go back a little bit to the control of the market, I could say that there are three pillars concerning medical technology. from the safety and effectiveness point of view. One is regulation that assures that only safe devices are coming on the market, and this is assured through the procedures that are already mentioned, the directives and the regulations in Europe, similar regulations in the United States by the FDA and in all other countries. There is a vigilance system as well associated with the medical device adverse events, and also HTA that assures that after certain period of use of the technology it is still effective, but there is no regulated the use of the devices. So it is not regulated that the user should know how to use the device, and this is something that is missing. Okay, I will continue a little bit. In Europe, just to tell you that this is a triangle, that it is the competent authorities that are controlling the market in each country, the notified bodies and the manufacturers, that it is the system in the European Union that is applied for putting on the market medical devices. Even if a device is correctly designed, manufactured, it is always possible to have some problems in the clinical practice. In many cases, some errors in the production line, some errors in the elements of the devices can produce adverse events in the hospital. And we are used now to have recalls, for instance, for cars. All the big firms, from time to time, they recall something, that something is going wrong to a car, and they recall them to change. It is exactly the same what happened with medical devices. If there is something wrong, and we have adverse event reports, then the regulatory authorities, FDA and the European Union, they asking the manufacturer to record the device and make the necessary changes in order to be safe again. And this is called the medical device vigilance system. The medical device vigilance system, it is based on reports from the users or even patients that they are saying that something went wrong with the use of a device. And if this is, let's say, serious, either severe harm or death of a patient, then the manufacturers, they have to go and make an investigation and probably go on with the recall of the device to make the necessary changes. This way, we avoid to have repetition of the same type of adverse events in the same or other places, other hospitals, or even at another time, similar devices from another manufacturer could have the same outcome. So, adverse events reporting system, it is very critical in order to better manage medical device safety. Just to give you an example, we had about 1,000 recalls in the early 2000s of medical devices. Today we have thousands of recalls. And what is more important is that FDA receives now more than 3 million reports of adverse events per year. And all this information is more and more let's say, triaged, I would say, through artificial intelligence systems that they identify the more important, and then they are going on with human interaction. Just this is in these graphs here, how this type of reports, adverse events are growing, and during the last 10 years, this is something that is continually growing. FDA has a very important database that has all the reports inside, and it is accessible from anybody. It is in public domain, all the reports, so we can go in and make studies out of these reports. In contrary, in the European Union, the corresponding database, UDAMED, on medical device adverse events, is not public domain, only restricted to the competent authorities, so a very, very small number of organizations in the European Union. However, we hope that this database will become also available to the public in the future. Some common cases of adverse events with medical devices due to use errors now. The more the devices are used, the more errors we can have. And errors could come with infusion pumps, for instance, in either the time or the way that it is administered, the medicine, with surgical robots, because surgical robots require a very well-trained of the staff, the clinicians, and also all the paramedical staff that are concerned. Ventilators, and we have seen that also during the COVID pandemic. We have seen ventilators that the staff was not familiar with the ventilators that they were using, and they made mistakes. We have also seen that clinicians trying to get more out of one ventilator for a patient, they try to connect two patients with a ventilator, which is completely out of any, let's say, practical way of trying this. And we have also communications and publications on the success of such trials. In just 2 months, the anesthesiology societies in the United States, they said, please stop. You can lose 2 patients if you put them together in the same ventilator instead of one. We have errors due to the incorrect use of electronic health records, blood glucose monitors, etc. And just to give you some data from one single hospital in Greece. If we go to infusion pumps, in this hospital we had an inventory of 388 devices, 13 different types. Each type of infusion pump is different, a different user interface, a different way, and nurses have to be familiar with all of them. If you change from one to another, errors are happening very often. It is exactly the same with ventilators, portable and transport ventilators. 17 different types. Similar with ventilators in the IC unit. Very different interfaces everywhere. Very different types of multi-physiological parameter monitoring system. 38 different types of bedside monitoring system. Now you understand a nurse or a medical doctor, a clinician that has to deal with different types of. We have one car, and if we enter to another car, we don't know sometimes where it is. The brake, the hand, or the brake for stopping the car, for parking the car. And now here you have to be familiar with all these things. It's also something that grows now very quickly, is home care. And in home care we have now hospital beds, patient lifts, wheelchairs, blood and glucose monitors, oxygen concentrators, or even a hemodialysis machine. And what is very particular in home care, we have not even professionals that are dealing with these devices. Sometimes are the relatives of the patient that are taking care. And we have much more cases of adverse events in this situation as well. Final training, it is important. We must train and have continuous education and training of the medical and paramedical staff of the hospitals, because in this way we ensure patient safety and by reducing the user errors, improve healthcare quality of the patients, exchange also enhance patient experience. They experience much better application of the health technology, and also by trained personnel, they observe what maybe is wrong, and so they can contribute better to innovation in the healthcare technology. I think that I will give you some examples here that happened with COVID-19, that in spite of the burden we have with this, the COVID-19 pandemic pushed a lot of things to go to the right way. And one of these was that we had a lot of training programs from all biomedical engineering societies and international organizations, like GCEA and IFMBE. WHO had a lot of such products and seminars and webinars, etc. We have also, as IFMBE, quite a lot of applications on the internet. So, technology is great, but should be used in a safe manner, not misused, not under or overused. Human factors and user errors are in most of the cases the root of many technology-related problems. Sometimes poorly designed equipment and technologies are the root of user errors, and vigilance reports that are reports of adverse events, I repeat, should be encouraged. We must also be prepared to respond to new type of adverse events with the use of new and emerging technology. One of them is AI, and continuous education and training is essential. Keep in mind that patient safety is a top priority, and to ensure we have to better manage medical technology in the hospital environment as well, and clinical engineers, coming back to the main objective of my speech, it is that clinical engineers are the engineers that in the hospital environment they are responsible for good use of and maintenance of medical devices, and so they are in the front line to make safety happen. Thank you very much.
Dear Professor Pankaratis, thank you for the very, very interesting presentation. So the next is open for discussion. Professor Fomin.
Thank you for your motivating presentation. And you mentioned this notion a couple of times, AI. This is a mark of changing paradigm in the clinical engineering. My question is, to which extent shall we, in general, rely on decision-making using AI? Do you see some border between immense help to use giant big data which is accumulated in your field, and very specific decision makers. Should it be left to us, or everything can be up to AI? Thank you.
Thank you for the question. I think that AI will be in our life in any case. It is already there. We cannot make a lot of things. But in the European Union, AI, application of AI will be regulated, and the new regulation is coming on the use of AI. Now, it is already as a clinical support, diagnostic support tool, it is used quite a long time ago, and it is entering into clinical engineering as well, because we have more and more data that are coming from the equipment, and this data can be used in order to see how, if we let's say, maintain the equipment, or we under-maintain the equipment, and so on. So sometimes we can predict problems that will arise in the future in the equipment and so on. So it is a very helpful and will be very helpful tool in our hands, but we have to use it correctly. This is the way.
Yeah, other questions, please. Your Excellency, please.
Thank you. Thank you for the very interesting presentation. This is a question and remarks not as ambassador of Japan, but as a student who is immensely interested in the globalization and how the world economy works. I took a great interest in one of your slides in which it said that the number of patent registration in the EU is 41%, that in the United States is 38%, and the rest of the world is 21%, right? And I found it very interesting, and well, I'm sure that the figures must be correct, but there must be something wrong or tricky in it, because it is impossible for me to believe that all the patent registrations done honestly by all the countries, including Japan, Korea, China, India, Canada, Australia, Singapore, etc., would be 21% compared with 41% for the entire EU countries. And now, that brought me to something that I read about as the tax, corporate tax evasion, in which companies like Amazon or Google make their tax headquarters in countries like Ireland, because by depositing their, by parking their intellectual property rights in those light tax countries, they can avoid being taxed in countries where they make money, but they can, attribute most of their profits into those, light tax countries, and can, prevent being taxed heavily. I wonder if the same, mechanism might be at play. And the fact that 95% of the small and medium enterprises the 90% and 5% of the medical companies are small and medium enterprises, which I'm sure obtain some intellectual property or patent rights. So if they register their companies in those light corporate taxing countries, then they can expect so my schools, they can expect to be taxed lightly and maximize profit. That way, European countries in which there are many light tax countries, can probably absorb so many patent applications and patent registrations. Am I right? Or if that's the case, nobody has written it. I follow globalization, and I haven't read it anywhere. But there is a big, there can be a big link between the European dominance in regulations in so many areas, including medical and safety designs, on the one hand, and the low tax status of countries like Ireland, Switzerland, British Cayman Islands, etc., and the Netherlands. I'm not criticizing. I'm trying to understand how the world works. But looking at those interesting figures, these I think assumptions and these doubts came to my mind, and I wonder if you can shed some light on it.
Thank you very much for the question. I think I understand that these figures are strange for you. It's not mine. All this information are not my research. It's MedTech Europe, which is the representative of medical equipment manufacturers, medical device manufacturers in Europe, that presents this in their statistics. So it are the figures that MedTech Europe provide for year 2021 or 22. A second comment is that 15,000 of applications does not mean 15,000 of licensing, but at least 10,000 of licensing applications that were successful are given, and from these 10,000, of course, no more than 1,000 will come to the market, probably less than a few hundreds will come to the market of these applications. Now, I cannot give you some more information on how MedTech is taking and put it in using in its statistics all the data that they have. But of course we have Europe, that it is composed by all the member states of the European Union, plus other countries that are not Swiss and Norway and UK now, that are not part of the EU, that are in these statistics. So I cannot guarantee that these figures are correct. I don't know exactly what is included or not included in the statistics, but at least I wanted to demonstrate that this is a very growing market, goes to 600 billion euros. If I go back 5 years ago, this was 250, 300 billions. So it's a very fast-growing market since the last 10 years. Many new applications means a lot of innovation in medical, in biomedical technology. And if I go back 20 years ago, probably in Greece we had 5 laboratories that they were dealing with medical technology, and now there are more than 100. All disciplines of engineering are now dealing with medical technology. So there is a proliferation of technologies, and let's say I don't want to stick on the correctness of the statistics. Are statistics that are published by MedTech Europe, but it is just a big figure that is important for us.
Thank you. Are there any other questions? Not, so Professor Korikarakis, thank you very much for your nice presentation. And Professor Fitonori Mimura, please go to the board.
So I'd like to introduce Professor Adli. So he participated every so this conference, and he made even interesting so the talks at the conference. So he's a really expert on the nanotechnology and solar material. So Professor Adli, so please.
Yeah, thank you. Thank you very much. Thank you very much. Is it working? Thank you very much for the kind invitation, and I'm very happy also at this conference to provide a talk. So I would like to stop you from getting lunch in time by giving a talk about aeromaterials. And would like to introduce our initiative for new antiviral agents. What you see here is one of our main players. I think we found out that this LED screen is unfortunately not reflecting the laser, so I do like this. So you see here this big block of aeromaterial, which is extremely lightweight. So this has just a density of 2 milligrams per cubic centimeter. So it's a funny nanomaterial. It has odd properties. For example, the van der Waals force is much more important here to this material than the gravitational force. This is why it's kind of floating on top of a box from PMMA. Okay, you will see this anyway later during the talk. So I just brought you an image from Kiel. Kiel University is maybe not well known. So Kiel is at the Baltic Sea, very much in the north of Germany. But I was delighted to be pointed out by the colleague from Japan that he said there was an uprising started in Kiel because this was a part where Germany started to get democratic after the First World War, and this was exactly at this shipyard you see there. The Faculty of Engineering, where I'm working, is right next to that. So I would like to talk a bit about the odd brother of gallium nitride. So if you're working in semiconductor science, zinc oxide is not taken serious. It's a 3,5 semiconductor, a strange one, because it has oxygen vacancies. Basically, from Ion Tiginano, we heard a lot about gallium nitride. So it has basically exactly the same structure, but it's not so inert. So it's not used in blue light emitting diodes, and it's also not used in power electronics, but it can do some things. For example, it can be used as a kind of biomaterial, and it's also antibacterial. So if you have zinc oxide nanoparticles, and if you put them, for example, into the kind of tooth pockets, then you can do dental treatments, for example, root treatments, in order to kill the right kind of bacteria there by an overdose of zinc. Together with Ali Kadem Hosseini, we made some patch which was added with stem cells, and this was implanted into rats for building to the one side gingiva and to the other bone, and that worked fortunately with very standard, and I would say the boring zinc oxide nanoparticles, just by an additional release of zinc. I was very intrigued by publications, for example, from Carsten Ronning, who was describing about a funny way how zinc oxide crystals can grow. So you can grow them, for example, in the shape of tetrapods we saw already before. So this is a self-organization, which is basically a very nice feature. And these are microcrystals. This is a small variant, but ours are usually much larger. How are we growing them? So we call this transport synthesis. So you have inside of an oven zinc plus a sacrificial polymer or ethanol in order to steal the oxygen, and after all this polymer or ethanol is consumed, you can then have a reaction of the zinc with the additional oxygen which comes then in, and afterwards it looks like it snowed inside of the oven, so you have all this crystal growth. And it's now material science to kind of tune that and scale that up. You see there's this ruler sticking inside of this thing which looks like snow. Basically, it is similar to snow because zinc oxide is also very polar and grows therefore very fast in these spiky directions like these tetrapods. And what is the motivation? But I will tell later, because this is indeed a GMP certified drug, and we heard now in the talk before how important it is by a startup company. So how is it looking now if you take an electron microscope? So you see here different variants of tetrapods. So could be ones which have sharp edges, could be some which have more roundish edges. You see they are pretty large, 100 micrometer scale is here. And we are doing a lot of science with this. It's always a pleasure to work, of course, with Professor Testemitanu. We had many publications on that. If you're interested in zinc oxide, I recommend a review by Yogendra Kumar Mishra. It's a little bit outdated, 2018, but to get into that, it might be useful. The problem is that there's too much literature on zinc oxide, so there are 50,000 papers in the last 15 years, and this was 2080, so probably it's 100,000 or exponential growth. So you find everything and the opposite about zinc oxide, which is also problematic. So this is Dr. Mishra, please consult him. You have variants where you have, for example, very thin arms. So typically the tetrapod diameter is 1 to 4 micrometer, the arm diameter, and then there's sometimes nanoscale connections possible. If you have such a material, you should do biomedical tests, because if you're synthesizing such a thing, you're immediately thinking of asbestos. And so you should check how it works. Indeed, the nice thing is you can even inhale these zinc oxide tetrapods, because zinc oxide, other than gallium nitride, is decomposed inside of the body, so in the lung. So from a pH of 5 already, making it slightly acidic, the body draws its zinc out there, and you get your 13 milligram daily dose of zinc. And here's some tests with various tetrapods, and seeing them in relatively standardized in vitro tests, there's a toxic dose and so on. And one can of course grind them down and see if the toxicity increases and so on. It's very low toxicity, and so it's very hard to kill somebody with zinc oxide. So sodium chloride is much better, for example. So how to use this, and sometimes getting ideas in biomedicine is good if you have no clue. So first I would like to tell a little bit from material scientist perspective about viruses, or nanotechnology perspective. It's a nanoparticle. If you look inside, then there's some program inside DNA or RNA, and then a lot of features around, and we all know these spikes outside since Corona, so I don't have to explain too much, I think, about that. And so the virus is only the program, and the viral particle is this, which allows the virus to float through the body. So, one virus I just showed is the herpes simplex virus, which is a severe problem. So if you see this, for example, from the World Health Organization, then it's an estimated 3.7 billion people have a HSV-1 infection, and almost 500 million have an HSV-2 infection. We might all know the HSV-1 infection. This is where you get these blisters, and the HSV-2 is even more nasty, because this is in the genital region, and it's a pain in the lower area. So how does the virus do that? So the virus is kind of entering the cell because of these glycoprotein spikes. There's a fusion which works like this. This is here from this Nature article, and there's a kind of binding, and then a trick that is inside of the virus, the capsid, is kind of going all the way to the cell core. And if it's in the cell core, then it's like being stupid and opening the email with the virus. So they read at the cell core the virus, and then it's programmed to do a lot of viruses which flood out here. And you know the result, that your lips, for example, look like this. You get the nasty blisters here. So I was asked, without much ideas, what could you do as antiviral agent, and you had just these tetrapods at hand. I was asking the American colleague who was kind of collecting a lot of materials, nanomaterials, for antiviral, if the virus would be polar. I had no clue which virus it was. And so you see here the surface of the zinc oxide, and I said, okay, if it's polar, maybe such a thing will happen. You bind one of these proteins to the surface of the zinc oxide, and this way immobilize it. So basically, you grab the viron by the glucoprotein groups, and yeah, so it's the same trick like you might know from a pub where you want to eliminate the flies going around, so they're just glued to the surface. So this was a very first try, and then the main author is IFMBE here. He was the guy who was demanding it. It worked. It was really nice to see that there's a reduction, for example, of the entry acid into the cells. And yeah, here you see something strange, which is called UV minus and UV plus. Because when he first said, yeah, it works, like immediately I was like, okay, I'm not so sure. Could you do the following? Could you illuminate the zinc oxide tetrapods up front with UV light, like you do with sterilization? And then tell me what happens. Because zinc oxide has an amazing property to have an autophotocatalytic process, which is enhancing the number of oxygen vacancies. And in such a manner, it's much more effective if it's upfront illuminated, because you have more oxygen vacancies. For me, it was a slight proof already that obviously this binding somehow works. There was some more evidence than later on in all kinds of microscopies, but also some cell tests. So then the whole thing went further, and IFMBE was asking his old PhD supervisor, Deepak Shukla, who's very much devoted to this virus. And he did then, 5 years later, some animal experiments. And then we saw the following: if you do nothing with this virus, then here after 14 days, the population dies basically down to 20%. If you treat it with a zinc oxide tetrapodal material, the mice were very happy. So this was totally unexpected. We thought maybe we see something slightly which might work here, which shows a little bit kind of difference between the two groups, but there was like parties. They did this 3 times with 20 mice, different concentration, and always there was kind of very healthy mice, even though they were infected with HSV2. So what they found out was what they could later on show in vitro. The zinc oxide is binding the viral particles, so these are fluorescent labeled viral particles. And now the antigen presenting cells have the possibility to move over the tetrapod and take up this virus and present it then and alarm the immune system. And that is a kind of trick. So the treatment here of the mice was at the mucosa, and that's where the viruses, the immune system, and these particles come together. So yeah, I don't understand all this biomedical stuff that well. They did some scores and took all the details. So I would like to forward you to this publication, which was then in 2016, and then there was a series of publications. Most done as last author by Deepak Shukla, and we were providing the tetrapodal material. And he could show then via these signal pathways, CD4, CD8, that it's really working and immunizing. So now, of course, the thing is how to get this stuff into the pharmacy. First you need a kind of production site. So there's a startup company, Firestone, which made their own clean room, and you see here all this zinc oxide which is produced under pharmaceutical regulations, which are very important to keep it absolutely clean. The good thing is that there is no kind of additional decision to make whether you take the zinc oxide nanoparticles, so to say the boring zinc oxide, or zinc oxide microparticles with these tetrapodal spikes. Because in pharmaceutical regulations, it's just zinc oxide, and so done is harmless. And there's a million of already medications which show zinc oxide in there, so we could use this vehicle, and you were saying like over and under treatment. So for us it was like a possibility now to bring this into the market as a cream, but not claiming the special effect, at least not in Europe. In the Arabian countries there's a sister product where you can claim it because of this animal study, but here it's not allowed. But typically people do not care if it works, and we have already some tiny studies, and that is now of course what we will do in a big study then, to see the comparison between the standard zinc oxide, which is already used for herpes, for example, for a little disinfection, and also drying it a little bit and so on, compared to the tetrachodal zinc oxide. And we had some found similar to the mice, some effects where people were immunized. So this was a very nice discovery which brought us quite into this market, and the company now had also some investments, was for this year done a couple of million euros for this upscaling. However, it would be a kind of funny thing if this would be the only kind of application, would have been strange. So we kind of considered that more. I just wanted to explain, I'm wearing this mask because I caught a light flu and I don't want to take these viruses where this stuff is not effective against to you. This exhausts me also a little bit. So as we had spontaneous emission of awards, I was saying maybe we can have a spontaneous income of speakers. So the further I would like to ask Dr. Siebert, Dr. Leonhard Siebert, because he did most of the stuff, to continue, and later on I hope I can take over for the last 5 minutes.
Bună ziua, my name is Leonhard Siebert. I'm a postdoc at Reiner's group, and I'm mainly working on these biomaterial things. And Rainer really nicely now demonstrated to you the biomedical capabilities of the zinc oxide, especially putting it into the screen. There are, however, many or other situations where a cream is not a sufficient vehicle for the treatment. For example, when you have a chronic wound in, for example, these food ulcers that appear in diabetes, then a cream is often not sufficient because you have a multifactorial problem here. You have reduced density of blood vessels, so reduced angiogenesis, no new blood vessels growing. You need some oxygen to dry out the wound, and sometimes it's too dry. You have not enough of the right growth factors to trigger the healing and so on. And most of the time the trigger for this are multi-resistant bacteria, not viruses. And since bacteria are a little bit larger than viruses, the same mechanisms for gluing it on the surface does not work. So what can we do there? We can, and this is my special expertise, use 3D printing of hydrogels and incorporate these particles into it so that they protrude from the surface of these hydrogels. This is very interesting because then on the one hand you have these particles really fixed in a 3D space. You can arrange these individual lines via 3D printing so that it stays nice and open, so the wounds can be oxygenated, and at the same time they come in perfect contact here with the wound and can have their special antibacterial properties. And what's more, you can even decorate the surface of these particles with some drugs or proteins to deliver it directly into the wound. This is how 3D printing of such systems work. It's not so super new to 3D print hydrogels. This is not the special part. The special part is really this incorporation of these large, complex shaped particles so they do not demix. They form natural filter, and it was quite tricky to get it so that you actually can print it. You can show it very nicely, however, so we can print geometries that are quite open structured. So on the one hand you have an occlusion of the wound, so no bacteria can come directly to the surface. And on the other hand you have high permeation, so you can get some oxygen there to promote the wound healing. At the same time, you can see here in these SEM images, the zinc oxide is protruding really from the surface of this hydrogel, so they are directly then in contact with the wound and can do their zinc oxide magic. Now the next step on top of this is you maybe want to use them as carriers to functionalize them with some of the proteins that help for wound healing. But it's, as we have heard already 2 or 3 times today, it's a semiconductor like gallium nitride or your zinc oxide nanoparticles. So it has very specific interactions with proteins. We found this out during my stay in Harvard, where we tried to really coat the surface of these particles with some fluorescence marked proteins. And you can see here, this is the normal fluorescence of the zinc oxide in blue, and the red part here is now protein that's coated on the surface, but it's not so perfectly coated everywhere. What you can do then is you can modify the zinc oxide on the surface. For example, here I just put it into hydrogen peroxide, then it turns into zinc peroxide, which is superpolar, and it can attract some of the proteins. But there's also other possibilities. You can think about the special properties that proteins have in solution, and that is that they have a so-called isoelectric point. That means at a certain pH value of the solution, the molecule will be neutral. Below that, it will be negatively charged, and above that, it will be positively charged. Maybe I'm saying it the wrong way, but this is a quite interesting property. Rainer showed you already these oxygen vacancies here in zinc oxide, and the interesting part about this is that in solution, or also normally they are charged negative. So if you really want to bind a protein to the surface of this zinc oxide, you should take care that it's in a positive state, otherwise it will be repelled. You can see this pretty nicely there on the top left. This is a logarithmic plot showing the protein loading, and the two proteins that we have put on top here, which are positively charged in pH 7, are the ones that we could coat the nicest, and the other ones have different size, but we had really big problems of getting them on top. You can see it here. This was too fast. You can see this here, there's some sort of phase separation that you see from this protein, which is not so nicely coated. On the other hand, all of this large black blob is one big chunk of protein that you can put on top. These are now just some very standard proteins derived from eggs and milk, I think, but it of course works the same way also with functional proteins like VEGF. The other thing that Rainer has demonstrated to you is that you can tailor these oxygen vacancies by just illuminating the zinc oxide prior to exposure to the proteins. And here we have the same protein solution. Top left is as received zinc oxide, and then we treated it in the dry state with UV light, and then we put it again in the same protein solution. Now everything is nicely covered, and you can use it then as a carrier for these proteins. We find this very interesting because it also means that it interacts completely differently with cells and viruses and bacteria, depending on the pH value, depending on what the cells do exactly in their neighborhood and so on. And one place where you can see this very often is if you try it in cell culture. So in cell culture, we can see that here at concentrations of already 3%, this is like 30 milligram per milliliter, both muscle cells and human blood cells are already dying, so a little bit toxic there. But on the other hand, in this mouse model and with skin explants, so ex vivo skin from real humans, we do not find any toxicity even up to 50% of zinc oxide. So this working together of cells in a compound here is of course very important for the resistance against the zinc oxide, which is something that the bacteria in these models do not have. And then from this, you can really go ahead and develop wound patches that you can use and apply to put it on wounds here. In this story, we have coated VEGF. This is a protein which helps in the regeneration of blood vessels, so forms angiogenesis. You can get it off of the surface of the zinc oxide with light because it's a semiconductor, and it shows great antibacterial properties against these classic hospital germs, Staphylococcus and Pseudomonas, which was really great. Yeah, we could publish this, and now I'm actually exhausted, so spontaneous phase separation again. Have to give back. Thank you.
Yeah, thank you very much for helping out here and staying in time. So this was this biomedical part. There's also another part which is basically triggered by the properties of collections of tetrapods. You can bring them together, you can sinter them, and in this way you have a very open porous flexible material. So here you see it's like a flexible ceramic. So it's like glass fibers which you can easily blend because they are much smaller. That is the basis for a lot of stuff. So here you see how we do this. You can take the individual tetrapods, sinter them, and come with kind of different type of shape. And this is now a good basis to do all kinds of things. We have seen how experts with gallium nitride can use this as a template and do gallium nitride, aerogallonite. And we could do the same also with carbon. This was a joint work when we discovered this with Professor Schulte, due to a hydrogen valve which was broken, fortunately the zinc oxide was etched away, and that was very nice, because then we could see that you can kind of make the whole thing porous. You see it up there in the tetrapod, you can look through even in a scanning electron microscope. So the wall thickness is extremely thin, and that material is still quite dark, because if light goes in, it's kind of scattered and absorbed sooner or later, even though the material is basically not there. So it contains 99.99% air, basically, porosity, and it's marginal there. Okay, we can play around with this. So I will show that even though the material is dramatically lightweight, it withstands quite some force. So in the upper movie you see how it's compressed and then unfolded again. In the lower you see how it's bounced around by electrostatics. So it's very interesting, but I have no time to explain in detail. The only thing is now it's in slow motion, that it's not easy to decompose the material. So in a kind of way it's robust. And you can also kind of heat it up by putting it between two contacts in vacuum and then treat it in such a way. And yeah, here's some more details, maybe I skip them, the literature anyway about the robustness of this material. Yeah, we heard one talent, maybe, hopefully, sometime soon to be professor, Dr. Siebert. There's another person, Dr. Fabian Schütt in my group, who's leading this team, and he was dealing great with infiltration of inks. So if you have transition metal dichlorogenides, or if you have graphene or whatever, you can infiltrate it. Like if you take sugar into a coffee, removing the sugar, then you have aero coffee, kind of. And so that, yeah, that is done here. And that is how the material looks like then afterwards. So the whole screen is maybe 100 micrometer, 100 to 100 micrometers. And yeah, that is basically where this material is composed out of. You can upscale it quite dramatically now. So this is 3 by 3 by 3 centimeters. And still the van der Waals forces are dominant. Here you can see it. So here one arm is removed. You see really it's very thin skin, similar like the aerogalvanite. And yeah, what can you do with this for biomedicine? This filter mask I have has one backdrop. All the viruses and bacteria agglomerate. But if you have a material like this graphene, it's conductive. And what you can do is, you can pulse, you can put some energy into this, like 5 volts, by pushing a current through. And as the material has basically no heat capacity, because it has no mass, it's relatively easy to achieve very high temperatures in very small time. So these were the first experiments. More than 4,000 Kelvin per second you can heat. It's more than 400,000 Kelvin. And afterwards the material kind of explodes. But before you're fine. So you can heat it up in a couple of milliseconds. So I think this movie shows this now, where you see it's kind of blinking, and you see it with an infrared camera on such a material. So the idea is you filter and you refresh the filter. So this was decomposition of pathogens, for example, for such a filter plate. Here you see again how the whole thing is kind of going on, and that you can kind of refresh your filter and then start off again. And yeah, the filter we were working on is a filter of an Airbus A320. This is a nice collaboration with Lufthansa Technik. So this filter is like that huge, which is mounted here in the lower compartment of the Airbus. And this is in the framework of the so-called graphene flagship, a spearhead project where a lot of companies, and again our startup company Firestone is working with. And it's nice that they got administration for aviation. I don't know if they will bring it into the market, but maybe for starting with some private airplanes or so, the people want to have the latest air filtration, maybe they will do this, but they were shown to be reliable in this way. Yeah. And if you heat the filter quite dramatically fast, then there was a discovery that the air as well is heated immediately, as you see here from that. So the air shoots out at the same moment while all the nanomaterial heats. Yeah, if you heat it in a millisecond to 400 degrees, simple P times V, pressure times volume, is proportional to the temperature, so general gas equation, and it's coming out and going back. And what you can do there finally with that is, for example, some actuation So you can do like a Carnot cycle, and besides the filtering, you can also heat the whole thing. Here you see you can also do it 100,000 times, it says cycle 100,000 times. And this is working as follows. So here's a tiny arrow material inside, where two contacts are mounted, and the whole thing is in nitrogen atmosphere now, into a syringe. And what you do now is pushing some current through, and you can heat it now to 1,000, 2,000 degrees without problems. And then all the air is heated inside, and the whole thing is now doing an actuation. So you see that the syringe is kind of going up. So this can be a kind of filtering device, but at the same time an actuation device. And the nice thing is you can put some load on that. So if you take now these 2 kilograms and put them on top of the syringe, you're compressing the air slightly, and then you see it's kind of still moving up. So it's quite powerful to its weight, because the weight is like a milligram, so which we find quite exciting. So this is still kind of on the way from basic research now towards application, and we can use this also for pumps. Here you see, for example, soft robotics would be imaginable. Here you see, for example, a rubber membrane which is pulsed and opened. So why can we use a rubber membrane even though we go to a couple of 100 degrees? Well, we do only with a tiny amount of air to this couple of 100 degrees C. So all the other heat capacity is so high that simply nothing heats up that dramatically fast. And you can see here, you can tune it. And yeah, these connectors can be shown here. Okay, what's going on? I have very talented PhD students, for example, Meta Haug and Lena Sauber, and they did now hydrogels as another variant, either with graphene underneath or arrow hydrogels themselves, and they can be utilized, for example, underwater for actuators. So this is a new work which just came out, and the nice thing is here you're switching light and you're not switching with electricity. So as a perspective, so this is a paper which just came out. Fabian Schütt, he's hopefully soon somewhere professor, if you think of inviting him over. He's really great. Yeah, you see him here. And furthermore, in collaboration, we had a nice collaboration with Testemitanu about the aerogalnite. Another variant is with Professor Faupel, who gives a plenary talk tomorrow, and Stefan Schröder, who has also a talk, who's doing some aero materials with hydrogels, and with some colleagues in Rostock, we could use them as cell templates. And that was very nice because he had cardiac cells, and this was now beating for a couple of months, which is very nice, maybe use of that. And you can either put the barring underneath or not with the graphene, so we are quite flexible. So I would be very happy if you could team up with one or the other person, if this is of interest for you, to make kind of special templates, or from the material science point of view, if this is of interest. I would like to thank my group, which is extremely productive and doing all that, and I would say new materials, new function, new demonstrators, and continuous innovation. Thank you very much for your attention.
So I think I go for discussion, so this is any questions. So you quoted the zinc oxide protocol and to kill the virus or the bacteria. So do you always need the UV light, or not always need the UV light?
The UV light is not that necessary for that. You can also do the oxygen vacancies in a different manner. So this process, for example, which is done by the Firestone company, already has built-in enough oxygen vacancies, and as it's immediately going into a cream, I have one if somebody is interested somewhere here. So it goes into the cream, then there's also very slow degradation of the oxygen vacancies. There's some kind of shelf life, which is exactly showing that. So, and there's also one thing, if you apply the, for example, to herpes blisters, then you're kind of also decomposing and breaking the tetrapods, which is doing, again, a kind of activation. You have then this high energy facets where the viruses are easily attached. And if the immune system shows up, so I was get rid at least of my herpes. We have some mini studies already which shows this in a significance, but of course a larger study should be now done, an application study, and then we can also in Europe and the US say, hey, this has an additional effect, just not being the boring zinc oxide. Thank you.
I showed some slides, and actually we have this kind of materials when they are organized, like fuse electricity and so on. We saw that in the homogeneous electric field, we have very pairs phenomena that could be used in nanorobotics. Have you thought about this possibility?
We have not yet employed the piezoelectric properties. That would be, of course, nice. We have done this now with the soft robotics, for example, with these type of illumination of the iron materials inside of the hydrogel. Then you're basically absorbing light, creating little temperatures. The hydrogels, which strangely, if they go over 35 degrees, they kind of shrink instead of kind of going. This has to do something with the inner degrees of freedom of these polymers, and then they're kind of like a sponge kind of being wrapped out. And in this way it was possible to do some self-actuators. But of course it would be very interesting also to study whether it's possible, for example, to do a polarization of something we put for purpose on that, and we can polarize it at once or something like that, that the whole thing has a kind of thing. Might be, I think we have anyway endless possibilities. I'm very happy to discuss with you in the future.
Any questions? No, it's okay. So, thank you very much.